their structures and predate our nematodes. And some fungi and bacteria directly attack and parasitize. Some compete for food and space and suppress population of nematodes. Some produce toxic compounds and directly kill nematodes. Among fungal antagonists, nematode fungi are most important. We have been studying since ages. They're very attractive and very interesting creativity and trapping mechanisms are there. But most of these are not practical and till now not been successful for farmers use. But in future there will be potential agents. They produce adhesive knobs, rings, nets, hyphae, and different very interesting pictures out there how to trap nematodes. But under field conditions, they somehow not results are not consistent. Some produce adhesive conidia also. And among fungi, we also have endozoic fungi, opportunistic fungi, AM fungi. But these opportunistic and AM fungi are most important, and some of them are very practically viable. Coming to bacteria, we have different groups. Most important is Bacteria penetrans, which directly attack nematodes. However, being an obligate bacterium, successful mass multiplication is not yet possible by this, but some companies have come up with this culture also. In future, this will become a most potential bacterial bioagent, and lot of studies have been made on this. Other group is different species of Bacillus and Pseudomonas, which produce toxins apart from increasing plant growth. And endophytic fungi and bacteria are most important nowadays, and they have been successfully being marketed and used by farmers because of the benefits like they control a wide range of nematodes and different modes of actions. They inhibit juvenile mobility. They prevent nematodes penetrating into the host. The most important mode of action is they interfere with giant cell formation. As you know, nematodes like root knot nematodes, they cannot feed on normal healthy cells. They need to modify normal cells into giant cell and then they feed. So without giant cells, nematodes cannot feed and multiply. And some produce toxins and kills nematodes. Some induce host defense resistance, and many they compete with nematode for resources and promote plant growth. Coming to fungal endophytes, the long list is there, but not all are important. We have two important organisms, Faconia chlamydosporia. This Faconia is the revised name for Verticillium. Purpurocillium liliaceae, revised name for Pacillomyces liliaceus. Among bacteria, we have different species of Pseudomonas, Florezens, Putida, and Bacillus. And we also have known as biofertilizer. Along with its regular benefits like enhancing resistance or nutrient uptake by plants, it also prevents nematodes in a mechanical way. The thick network of hyphae and mycelia and chlamydospores of these mycorrhiza, they colonize roots, particularly xylem and phloem vessels, and they prevent the entry of juvenile of nematodes, and also they prevent in the free movement and multiplication of nematodes. This is a very good option for farmers. Among many genera, Glomus fasciculatum, Glomus nosae are most important. US Bangalore has developed a technology also at 50 grams per square meter in acidic beds of tomato and brinjal. It effectively manages root knot nematodes, and each gram should have at least 200 chlamydospores. This is a colonization of roots with chlamydospores and mycel hypha. P. liliaceum is the most popular fungal bioagent available today, and farmers they use it profusely on open field and foliose crops, mainly because of its benefits like it attacks nematode eggs, adult females, different stages of nematodes. It can grow on or inside the nematode and also secrete enzymes to weaken nematodes. Shrinkage of eggs, killing of developing juvenile is a major mode of action. It directly attacks adult females of nematodes, root knot nematode particularly, and forms thick network of spores and mycelia on the cuticle. Faconia chlamydosporia is the next best option for fungal bioagents. It also attacks eggs, colonizes saprophytically, endophytically, and also induces plant defense mechanism. Important mode of action here is complete death of developing juvenile, immature death of juvenile, and also shrinkage of eggshell. Trichoderma viridae and arsenum are also on the popular list, which are used by farmers. They have different modes of actions. Main mode of action is directly toxic compounds like viridin and trichodermin. They kill nematodes, and they produce different enzymes to react and kill, destruct cell wall of nematodes. They also compete by utilizing protoplasm as a source of food and multiply spores. Among bacteria, P. penetrans, as I told, it's a very important potential bacterium. Its endospores directly attach to the cuticle of nematode, germinate, enter inside the body of nematode, profusely multiply, and rupturing of cuticle of nematode occurs. This is a sure killing of nematode. Reproduction inside the nematode body. This is the attachment of spore on the cuticle and killing of nematodes. We have a technology at COH Bangalore, nursery treatment of tomato, brinjal and rice with pea penetrance, 20 gram per square meter, effectively minimizes population of nematodes. Various species of Bacillus are very popular, and most important ones are Bacillus subtilis, and it's very effective against a wide range of nematodes. It colonizes roots and with multiple modes of actions like acquired immune systemic resistance, stimulate phosphorus solubilization, and produces various cell wall hydrolytic enzymes and antibiotics to kill nematodes. Its main mode of action is it forms a thin colony around the root zone so that second stage juveniles of nematode cannot reach the root and they cannot find out a feeding site to penetrate inside. So its repelling action is there. And Bacillus thuringiensis is the most popular against insects. It also works effectively against nematodes. It produces delta endotoxins, thuringiensin, which mainly interferes with RNA transcription by inhibiting RNA polymerase. And after treating, nematodes exhibit various anomalies, the treated and untreated nematodes with the crime. Ivermectin is one of the important upcoming popular bionematicide. They are macrocyclic lactones derived from mycelia of Streptomyces ivermectilis. They are anti-nematicidal. They mainly block neurotransmitter GABA. One of the most popular commercially viable ivermectin is abamectin. It's being marketed. And other brand names are ivermectin, selamectin, and doramectin. Pseudomonas fluorescens is the most popular running bacterial bioagent at present. It has many beneficial effects as a PGPR, systemic in nature, and it completely destroys the cell wall of nematodes by producing enzymes, toxins, and other byproducts. And also inhibits growth of nematodes. Development of J2 into adult is inhibited with various antibiotics and induce systemic resistance. Most beneficial effect of all these biogens are they have a very types of methods of application. You can broadcast furrow application, resistor application or root zone application, seed treatment, wound application, or to control foliar nematodes, you can spray onto the plant or you can spray onto soil surface. However, most popular methods are resistor application and broadcast methods. And when we say biogens kill nematodes, you should know how exactly nematode is killed and how it is shown. Mostly, most biogens, they inhibit hatching of eggs of nematodes. Without hatching, no second stage juvenile, no infection. They also inhibit molting, developmental stages are affected, directly kill the juveniles, so adults will not be formed. And most of them, they affect sense organs, which make nematodes to fail to search for a host in soil or to search for a mate for reproduction. And overall growth of nematode is also affected. Some bioagents, they affect fecundity of females. Root knot nematodes average number of eggs laid are 500, 600. If it is treated with bioagent, its number will be reduced to 15 to 20 eggs. So they also infect or affect hormone metabolism, imbalance will be there. And some bioagent stimulate host to produce more of nematicidal metabolites as root exudates. So such plants do not attract nematodes. They have a repelling action of nematodes. Some pictures showing the effect of bioagents on distortion of eggs, eggshell, and killing of developing juvenile. And below picture is the control with healthy developing juvenile inside the egg. We all know that bioagents should be applied along with organic amendments. In India, most common organic amendments are farmyard manure and oil cakes, green manure, vermicompost. In oil cakes,
Ravichandra, your
yes,
time
sir?
is up.
Yes, sir.
Ravichandra, please
Yes,
conclude.
yes, yes. Okay, sure, sir. This is a list of fungal nematicides or bacterial nematicides, different companies in India and different countries, and many ISR institutes, national, international institutes, state agriculture are producing these bioagents. However, commonly used ones are these P-lilacin, trichoderma viridae, and Pseudomonas fluorescens. And consortia also is available. Farmers are preferring this consortia of various combinations, specific dosages, and pictures are treated and untreated ones with profuse root growth and number of gallings are reduced. And most important message is that a lot of fake products are available. We should encourage farmers to select only for genuine products to control nematodes. Thank you.
Thank you. Thank you, Ravichandra.
Thank you, sir. Thank you, sir.
Very quick, 1 or 2 questions, please. Otherwise, we are running short of time. And meanwhile, I request the second speaker, Professor Nina Mitter, to be ready. I can take 1 or 2 quick questions, please. Audience.
Ravichandra sir, excellent
Yes, sir.
presentation, sir.
Thank you. Thank you,
Actually,
sir. Sorry for some disturbance.
Your views on Vellum Prime and Nimitz, sir, please.
Yes, we have evaluated both the products. Between those two, Vellum Prime is the most effective against nematodes both in open field and polyos crops. But its dosage is also very less, 0.3 or 0.5 grams, I think. This comes next to Vellum Prime. Under heavy infestation of nematodes, Nimitz may not work properly. Vellum Prime is best. And low population, average population, Nimitz is okay.
Thank you.
Carbofuran being banned shortly, so which you strongly advise, sir?
Which one?
No, in place of carbofuran, which is going to be banned, right, sir, from now onwards.
Okay.
Most ideal method considering the environment and ecology.
Yes, yes. I think we cannot replace carbofuran because of phytotonic effect and also safety to other things. Efficacy-wise, Vellum Prime is better than Nimitz, and carbofuran can be replaced with Vellum Prime, not Nimitz.
Thank you, sir. Thank you very much.
Thank you.
Thank you, Ravichandra.
Thank you, sir.
With this, I invite the second speaker. Is she ready, madam? Professor Nina Mitter, are you ready? Professor Nina Mitter? Hello? Professor Nina Mitter?
Sir, I think Professor Nina Mitter is not there.
Okay.
Can we go to the next speaker, sir?
Okay. I request the next speaker, Nakiran.
So,
Elmira Yanti. Elmira Yanti.
Yes, I am. Yes, sir. Hello. Yes, sir. I'm Yulmira Yanti. Yes, sir.
Go ahead, madam.
Sorry, sir. I don't listen. Sorry. I don't hear you listen.
Hello.
Hello, hello.
Can you hear me, madam?
Yes, I'm Yulmira Yanti.
Okay.
Dr. Yel, please. Right.
Okay.
Just to introduce madam, she is from University Andalas, West Sumatra, Indonesia.
Yes.
And she is specialized in bacterial control of wilt.
Please share
She
the
has
slides.
21 publications and guided 7 PhD students. Now, madam, please go ahead with your presentation.
Okay, thank you very much.
Yes.
This is the presentation. This slide. Okay, thank you very much for your organization. I'm Yulmira Yanti and from Andalas University. Today I give you the title Development of a Compatible PGPR and Cyanobacteria. Cyanobacteria biocontrol agent for effective management bacterial wilt disease and Bemisia tabaci in tomato. Tomato Solanum lycopersicum, according to the FAO, is the second most cultivated vegetable in the world and make it none of the most economic important.
Madam, can you please do so?
Hello?
Can you please click on slideshow?
Okay. You see this slide show? Okay. Hello.
Not yet, madam.
Okay.
Ma'am, we could switch. Yes, continue, ma'am. Thank you.
Okay. Tomato Solanum lycopersicum, according to FAO, is the second most cultivated vegetable in the world and make it one of the most economic important crop. Bacterial disease. This bacterial will disease caused by Ralstonia syzygy subspecies indonesiensis, former name Ralstonia solanacearum, caused by a considerable amount of damage to tomato and many other crops. In the world, the pathogen in dispersal and one of the economically important bacterial plant pathogen. Management of bacterial while of tomato was difficult due to the soil-borne nature. Of the pathogen, pesticide used as control were not considered to the long-run solution to the concern of expand export resistant to toxicity known to target organism on the health environment hazard. Research study had been focused on developing eco-friendly, safe, and objective agent of plant pathogen and disease control option such plant growth promoting rhizobacteria (PGPR) and the novel strain of PGPR from cyanobacteria control which bacterial against such PGPR and cyanobacteria are not promoting control. Potential biocontrol agent, plant growth promoting rhizobacteria are potential agent for biocontrol of plant pathogen and well known to increase plant growth and crop productivity. Common mechanism, PGPR to promote growth of plant, clonates root tissue, increase nutrient uptake, produce phytohormone, produce active molecule, induces systemic resistance. Cyanobacteria, blue-green algae are not candidate biocontrol agent to enhance ground nitrogen fixation and yield in the cropping span. Cyanobacteria are eubulcation group of organisms which have been relatively less investigated as PGPR, although their role in nitrogen dynamic and paddy pest cropping system is well investigated. Cyanobacteria are not to produce comments, which range activities including phytohormone, biological, metabolic, or nutraceutical. Cyanobacteria role as PGPR found to be similar with the bacteria. The inoculation of plant with bacterial mixture consortium apparently provide greater benefit to plant growth than inoculation with single bacterial strain. Why consortium? This why consortium. This prospect use biocontrol agent as consortium. Microorganism is nature acts together consortium and more similar to ecosystem. Consortium of bacteria interacts synergistically can give bacteria that compared to single strain. Bacterial consortium provide more various control mechanism simultaneously. Study object, the plant work understand to base effective consortium consistent of plant growth promoting rhizobacteria and cyanobacteria through growth rate of tomato and control Ralstonia syzygy subspecies indonesiensis and Bemisia tabaci. The study was conducted in February-December, and the microbiology experiment Faculty Agriculture Andalas University Indonesia. The base consortium potential strain SI using cross-culture implanta assay for consortium ability to both control Ralstonia syzygy indonesiensis and Bemisia tabaci and to promote growth and yield. The base consortium. Strain were test the antagonistic for microbial consortium development following the method of cross culture in each different strain. The feature PGPR concern were development-based compatibility of the strain. Next potential cyanobacteria strain. The strain used this study as lies to the screen where the isolate promote growth tomato from the previous study. This PGR strain, Bacillus thuringiensis, Bacillus cereus, Bacillus cereus, Bacterium GV, Bacillus sp, Bacillus cereus GN23, Bacillus subtilis, BSN RBI IPBL, and strain Cyanobacteria RZ AB2.1, RZ2 AB2.3, RZ 1BPL 2.3, RZSG 1.1. This is my colleague Yanti. Consortia inoculant each of strain were growth sporadically using common methods of bacteria and cyanobacteria multiplication. Consortia made by mixing the compatible strain. This consortia this mass compatible and inoculated to seed by dipping for 10 minutes and planted in seed bed. This growth this tomato. Inoculation, pathogen inoculation, the Rasonia CZGE Indonesiensis strain were inoculated by pouring 10 milliliter of RSE to the damage by cutting root of tomato 2 weeks after seedling planting. Tabasi introduction. Bemisia tabasi were introduced by placing 1 male and 1 female of adult Bemisia tabasi to the consortia inoculated plant, then covered with screen nets. The Bemisia tabasi population were decimated until the last observation time. Parameter observation: plant growth, plant height, number of leaves, freeze flowing time, yields, pest development, disease incubation, disease incidence, TBC population. Result compatibility of PGPR and cyanobacteria strain. This is the table compatibility PGPR and cyanobacteria. This is compatibility C3. This is C3, C4 compatible R5. PGPR and cyanobacteria consortium decide for treatment Bacillus thuringiensis strain RB2 AB 1.1 Bacterium GP Cyanobacteria. C2 Bacillus thuringiensis strain RB1 2 AB11 Bacillus cereus strain APS SB 03 RBI 2 AB 2.2 and Cyanobacteria RZ2 IB 2.1. This code, please you see the strain code strain PGPR and Cyanobacteria Dimes consortium. Growth promoting activity of the PGPR and Cyanobacteria consortium on tomato. This plant height is consortia C2 effectivity 2.61, leaf number 62 and 60. This is freeze flowing time. This is consortia C2. This is the picture consortium C2 left and control right. This is consortia the big and the best the picture. Consortium ability Eson table strand compatibility with other strands, some strand and compatibility that were used to make compatible social for the study. This consortium ability as biocontrol agent Lawsonia CZGE subspecies indonesianensis on tomato. This consortia, the best consortia C2, C1, C3, C4, C5, and C6. No, there is no street, no live spot. The effectivity to act 0.04. In 0, this is incidency. Tomato inoculated with consortium and control while the right. Bemisia tabasi population each plant. C2 they act 7.667 in star 1 names 6.33 and in star 1 name 4.00 in star 2 name 1.667 and pupa 0.667. This consortia C1. This at 7.00 insar 4.333 and insar 1 0.667 and instar nip 0 and Papua 0. This control this certain instar 1 name 12 instar 1 name 9.33 and instar 2 name 7.33 and pupa 5.667. Memesia tabasi attack intensity consortia this control the height attack intensity different for consortia C7, C10, C11, C6, C4, C5, C1, and C2. Conclusion: the application of VGPR and cyanobacteria consortia be effective in controlling the bacterial wild disease Ralstonia CZGE subspecies indonesiensis and control Bemisia tabasi. The PGPR combination of C2 Bacillus thuringiensis strain RBI 2AB1.1 Bacillus cereus strain APSB 03 RBI 2AB 2.2 Bacillus subtilis BS 5 RBI IPBL 2.3 and cyanobacteria RZ 2AB 2.1. C2 Bacillus thuringiensis strain RBA 2A11 Bacterium GP60 RBI2 AB 2.3 Cyanobacteria RZ3 AB 2.1 RBI2 AB 2.3 Cyanobacteria RZ2AB 2.1 were the promotion consortia for the management of wild disease and Bemisia tabasi and enhance the growth of tomato plants. Instrument, thank you. The author will explain growth to Universitas Andalas for finding this research. The research percepatan guru besar in Indonesia scam with contract number 21 since March. Thank you very much for your attention and thank you. This I come to Mr. Okay, thank you. Okay, thank you.
Okay, thank you.
Thank you, madam, for a nice presentation and covering within the time limit. And let participants please, one or two quick questions.
Hello, madam.
Hello.
Dr. Yamira, you can close your PowerPoint presentation.
Okay, okay.
Madam,
Thank you.
Any questions, please?
Sorry, di Indonesia there's problem. There is a signal. Hello, there is my country there is problem. The signal.
Okay, sir, you can go to next turn, sir.
Okay. With this, we
Krishna
thank
sir.
Dr. Elmira Betri, and may I request Dr. Nakiran. Nakiran, please, you are ready?
Sir, Krishna sir.
Sir,
Huh?
Sir,
Krishna
Krishna
sir,
sir. One submission, sir.
Meter is here, sir.
ಹೇಳಿ ಒಬ್ಬರು ಮಾತಾಡಿ. Yeah.
Sorry, I don't listen to your speak for me. Sorry, I don't listen. Hello.
Okay. Thank you, madam. Thank you. Dr. Nakiran, sir.
Krishna sir.
Thank you, sir. Yes, sir.
ಹೇಳಿ.
Krishna sir.
ಹೇಳಿ.
Nina meter is there, sir.
ಇದಾರಾ?
She joined.
Okay.
Yeah.
So let me welcome Dr. Nina Mitter. Professor Nina Mitter, are you ready?
Yes, thank you. I am. Can you hear me?
Yeah, we can hear you.
Thank you.
We just introduce the
Yes, ma'am, we can hear
speaker. To introduce the speaker, she is from the Center of Horticulture Science, Australian Institute of Bioengineering Nanotechnology, and she has recognized several received awards, including the Prime Minister of India, Queensland International Fellowship, and several others. Madam, can I request you to make a presentation?
Thank you very much. Thank you. I'll start now. I'll
Yes,
start
ma'am.
sharing the slides. Let's see. Hope it works. Trying to share. Can you see the slides? Is that visible? The slides are visible?
Yeah.
Okay, thank you. Thank you very much. Thanks to Professor Devappa and all others who have organized this wonderful conference, and to the chairperson, Dr. Krishnappa, as well. My apologies. I have been trying to link, but I think there were server issues which were finally resolved. So thank you very much. It's always a pleasure for me to give a talk, wherever possible in India. It sort of brings back so many old memories, and I'm one of you. I did my PhD from Indian Agricultural Research Institute, and very much still linked to a lot of researchers in India. So this is once again a really thanks for the wonderful opportunity. I am, as was mentioned, I'm at the University of Queensland, and I'm the Director, Center for Horticultural Science there. The talk that I am today going to give is on RNA-based biopesticides, and especially looking at the theme of Sustainable Development Goals of sustainable crop protection. I don't think so. The whole audience is very much switched on. We all know about the issues of food and nutritional security. I think there is some background noise. So if people, I can hear some
Unmute.
background speaking. So, yep, thank you. Thanks a lot. So as I was saying, food and nutritional security issues are nothing new to everyone here. We know that 10 billion people will be walking on planet Earth by 2050. And it's not just about tomorrow,
Mallikarjun, please mute. Mallikarjun, please mute. Mallikarjun, please mute your microphone. Thank you.
Thank you, Dr. Krishnappa. Thanks a lot. Even today, we know that 66 million primary school children, may go to class hungry. Not only that, this pandemic has brought some very stark figures up front. I was reading that the FAO report that they were saying that 135 million people were below the chronic hunger line. But when the pandemic has happened, this number is going to double by the end of 2020. So this has really sort of brought to the forefront how important it is to have a good agri-food sector, a strong and vibrant agri-food sector if we need to meet those goals. And one of the ways is to sort of reduce crop losses. However, with or without pesticides, and pesticides are important, even when we use pesticides, we get losses of about 40% happening. But if we are looking for new safe chemistries, it's a very interesting stats that it may take 13 years and $139 million to research, develop, and register a new crop protection product. And only maybe 1 in 139,000 chemicals tested makes it from the laboratory to the farmer's field. And then we have to take into account some of those issues. There is an issue of resistance development, and we know that pathogens and pests are really clever. They can beat us anytime, and they do evolve and cause those losses. We also have the issues of residue. So we do not want any residual toxicity on our food crops. We are also aware of the issues of runoff to our precious waterways. Lack of specificity is another issue. We don't want to kill our beneficials. We don't want to kill our butterflies and bees, full insects. And as I mentioned, new chemicals are hard to come by. And this is what brings me to RNA-based biopesticides, which are based on this technology called RNA interference. And RNA interference, it was heralded as the most important thing to happen in molecular biology in the last 10 to 20 years. And I'm sure those of you who have read the news today, the Nobel Prize this time has gone to 2 women, which is really nice. So it's in big stars, big thumbs up for female scientists for the CRISPR-Cas9 or the gene editing technology. So this was really a good news across the globe that science is getting that quantum and recognition. So I'm talking about RNAi here, and I'm sure the audience would already, know about this, that normally when everything is genes are expressed, it is DNA, single-stranded RNA, and that. However, in the case of silencing or RNA interference, this DNA, instead of forming a single-stranded RNA, forms a double-stranded RNA. And this double-stranded RNA is the trigger. Plants have these scissors in their pocket or dicer enzymes, which they use. Plants are not multicultural at all. They don't like this foreigner dsRNA, and they chop it up into small bits called small interfering RNAs. So as you can see here, there is no protein. That means that the genes are not expressed. Not only that, these small interfering RNAs can dice up messenger RNA with whom they share a homology or a complete sequence homology. So this actually, when RNA interference came into the force, this heralded the era of developing transgenic or genetically modified plants, which have you integrate the genome into the genome so that they make the double-stranded RNA targeting the specific pathogen. We are all aware of the issues with GM, whether it is community acceptance, regulation of use, and cost and time involved. Can you hear an echo for me? Am I sounding okay, or is there an echo?
There is a slight echo, ma'am.
There is a repeat echo.
Unmuted the microphone. Ravi Kumar Naik. Ravi Kumar Naik, unmute. Thank you.
Mute, sir.
Mute, please, yeah. Shankarappa, mute. Shankarappa, please mute.
Yes, sir.
Yeah, thank you. Others also, please mute.
considering the issue of genetic modification and its acceptance, the idea that we started exploring about 5-7 years back was, can we deliver this RNA or double-stranded RNA as a spray on the plant? That is, as a topical application instead of modifying the genome of the plant. And whenever you think of those ideas, so I thought team thought of this idea way back in about 2011-12, and we were thinking we are really the first ones to think about it. But then you go to your Google friend and you start looking at the literature. It was Stenlado et al. In Spain, actually, for the first time, who used this idea in 2003. And this slide, you can see half of the leaf that has been sprayed with the double-stranded RNA targeting this particular pepper mild mottle virus, whereas the other half has not been sprayed, and then both halves were challenged with the virus, and you can see that the dsRNA spray protects. Since then, there have been lots of papers and publications coming out. I review almost 2 every month now. It works. It looks like that double-stranded RNA as a spray can work. And I was thinking, then what is the issue? This should be really a game changer. But then when you read those publications, you look at the end conclusion section, and it is always that this naked dsRNA, when it is sprayed on a plant, is highly unstable. It degrades on the plant surface. It can easily get washed off by rain, UV, sunlight, enzymes. So the protection window is very short, like it is 3 to 5 days. So if you spray your dsRNA on day 0 and your virus comes on day 5, the plant is protected. But if the virus comes on day 10, your plant is not protected. So therefore, this is not a farmer-friendly, viable solution to spray naked dsRNA. And this is where we started thinking that how can we make this dsRNA stable? Can we make it last longer? Can we have a slow release on the plant surface so that we can increase the protection window? And this is where we developed the concept of RNA-based biopesticide. And very pleased to say that we have developed this world's first technology called BioClay. BioClay is just as the name indicates, bio is RNA as the biological active ingredient, and clay particles are the nanoparticles that we use as carriers for this active. So clay is just like a fancy postman, and RNA is our biological active, but this clay provides those features of stability and safety. This clay is a really natural clay. And also we address the issue that it is completely biodegradable, so there is no residue left. And this particular work was funded initially by Bill and Melinda Gates Foundation. Very pleased to say that starting with $100,000 as a small proof of concept work, we are now $30 million investment into this platform, and it is to commercialize. So what is to deliver RNA? It can be applied as a spray application. Clay layers, they just degrade on the surface. It's almost like nature versus nature. So you look at your virus, you pick up a critical gene in the virus or an insect pest or a fungus, and you make double-stranded RNA for that particular gene sequence. And all then you have to do is load it onto the clay and spray it on the plant. So there is no genetic modification involved. This slide sort of captures the schematics of BioClay. As you can see here are these clay-loaded particles. We spray it on the leaf, and after that, it's a very natural process. In the presence of carbon dioxide and moisture on the leaf surface, this clay slowly degrades and releases the dsRNA. This dsRNA that enters into the leaf, and then that triggers the RNA interference pathway. We published this work in Nature Plants way back in 2017. As you can see here, yes, it really works. So if you look at this slide, this just is a blob showing, if you look at this panel with BioClay, even after 30 days of the spray, we could detect the double-stranded RNA on the leaf as compared to naked dsRNA, which disappeared at about 10 days, it started to degrade. We could show these are just fluorescent dsRNA in panel C. We could show that yes, the double-stranded RNA enters into the plant system. In panel B, we showed that it does not get washed off. So this is bioclay treated leaf, which is there, and it is after washing vigorously with water, and you can still see the fluorescent dsRNA. And panel D, I reached out to Francis Tenlado, got the same dsRNA that he had used for the virus control in 2003, where he could get a protection window of 5 to 7 days. And we could see that by using bioclay, we had extended that protection window to 20 days at that time in 2017, but now we have increased it further. So it really works. As I was saying, this is another virus that we have been working on, tomato spotted wilt virus or TSWV, and a range of tospoviruses actually. And here you can see a capsicum plant that has been sprayed with bioclay and challenged with the virus compared to a control plant. So it does give us nice protection. We are also now looking at insect pests. So we are targeting aphids and whiteflies as well, because we know that if the dsRNA is there in the plant system, the insects can suck up that dsRNA. So it will be insect targeting double-stranded RNA. The next step for these RNA-based biopesticides is we were looking at, can we control fungal diseases? So starting with a humble beginning with viruses, we have branched out to insects and now looking at fungal diseases as well. Now, this is Professor Hailing Jin at University of California, Riverside, and she's our collaborator. Here you can see that this is, if you see the last panel, this has been sprayed with double-stranded RNA targeting botrytis. And this also provides protection. And this opened up a new window for us because this means that we could use it for post-harvest as well. There is a possibility. I'm not saying that we have done it, but we are exploring that possibility that, yes, you know, this particular technology of RNA-based biopesticides using clay could be helpful maybe in post-harvest control as well. We are also working with Professor Kogel, Karl Kogel, in Germany, and once again he has shown that yes, you know, the concept works for fusarium on barley, and this is these assays, and this is the top panel which has been sprayed with a fusarium targeting double-stranded RNA, whereas these leaves have been sprayed with either a controlled dsRNA or no dsRNA at all, and you can see the difference in the symptoms. There is another group in Canada where they have done for sclerotinia and botrytis on canola. So once again, Steve Wiard's group and Belmonte's group have also shown that naked dsRNA applications can work. And we are very interested now in seeing whether bioclay can actually take it to the next level. So as in sort of doing this work, the story has been that yes, you know, we've been trying to develop this space of RNA-based biopesticides, and it has now resulted in a big hub now. What I'm leading as a director there for an additional hub, where Australian federal government, we have got about $18 million in cash and in kind, and there's various universities now involved. And the purpose of this hub is specifically to target fungal diseases of grain crops as well as horticultural crops. And in this hub process, because it's a big grant, it's not like a small project proposal. We are looking at right from design to laboratory trials to glasshouse to taking it to the field. Not only that, we are looking at getting insights into the mechanism, but scale up in terms of formulation development so that we can end up with a bioclay product. We also have a social licensing theme here to see, to work on the adoption of this. No technology good if the farmers are not going to adopt it. And we must respect their questions and we must make them involved in the process when the project or when the product is being developed, rather than developing the whole product and telling them, yes, how good it is. Now, I think I have really huge respect for our farmers and growers. I always feel that they know so much more than us, and we do need to learn from them. And if we make them as our partners in the beginning in the project, so growers are our partners in this project, they will be offering the seeds for the trial, and they will be the ones who will help us in making this product a reality. I must mention, we don't have the product on market or anything. I know I get this question asked after talks. We have a commercial industry partner. They are looking into the manufacturing and product development, but we do not have any commercial product at present. We are also trying to develop collaborations with India in this space, with various other organizations which are located here. So if I look at it, for me, this RNA-based biopesticides does present a paradigm shift because pre and post-harvest application is possible. It is pathogen specific. And the most important thing is, as I was saying, pathogens are very clever. They can develop anything, anytime. They can come and break and evolve or mutate. If you have a genetically modified plant, then you will have to start the process all over again of making a new transgenic line. In this case, even if the pathogen evolves or let us say mutates and your dsRNA is no longer effective, it will just take you 1 hour to design the new dsRNA sequence. All you have to look at is the genome of the pathogen and see where the mutation has happened. If the mutation has happened in your region of dsRNA target, you can design. And it is, as I was saying, drawback is that GM is for the life of the plant. So that's a big drawback here in case of RNA-based biopesticides. If you have a genetically modified plant, then you have protection for the life of the plant. In this case, it will work like any other fungicide or insecticide, that it will provide a protection window only for a limited period of time. So I am looking at this technology as something which we can add to our IPM programs. I'm not looking at it as a replacement totally of your fungicides or insecticides or pesticides, but this could be a really valuable addition to reduce the use of insecticides and pesticides. So this is something to take home. And we are also looking at it's with any technology, you have to acknowledge the risks, and we are looking at it in terms of dsRNA persistence in environment, in soil. However, at present, all the papers that we have done, and I am also a representative on the OECD committee, European Commission committee, where we are talking with all the 26 European countries, developing the regulation policy for the RNA-based biopesticides and addressing some of the concerns as and when they appear. But at present, there is no issue because double-stranded RNA is actually present in almost all plants, and we do eat them. Even if we are eating a latent virus-infected plant, it would have the dsRNA of that virus in some shape or form. So that's something that we look at. Very pleased to say again, this particular has actually resulted in Office of the Gene Technology Regulator in Australia declaring topical RNAi as non-GM. So when I started the work in 2013-14, only I was saying it is non-GM. So that was okay. And the other academics and other scientists were saying it's non-GM. But the legislator also has to say that it is non-GM. So we tabled the case to our Office of Gene Technology Regulator in Australia, and on 8th October last year, they passed the legislation that any topical RNAi is considered as non-GM. So we do not have to go for deregulation. From a GM perspective, we'll still have to register it as a biopesticide with our pest management agency, but not go for the deregulation of the trial, so which was a big win. As I said, I am really involved in nano-enabled agriculture and have written 2 pieces for Nature Nanotech recently. Once again, I am very conscious of the fact of policy and regulation. So when you look at a holistic way of any technology, you can't just look at the science and just push with the science. You have to look at the policy, you have to look at the regulation, and you have to look at the social side as well. And I think if we have all those concepts at the beginning of the project and make industry as your partner as well. So our industry partner has been there with us for the last 6 years. So they are more or less like research partner with us now. And that is what takes us to the next level. So yeah, I would like to end by saying endless possibilities. That's what I see happening. So we are working on viruses, fungi, insect pests. I see a huge benefit of this technology, especially in controlled environment cropping or covered cropping or protected cropping, whatever you call it, in greenhouses, because that is where you cannot use very toxic pesticides or insecticides. And this is where this biological alternative may have a more advantage. We have also now branched out from plant health into animal health as well. So we are looking at things like flies and ticks of animals. Not that I'm an animal scientist, but I have people who are doing that. So I just want to stay on the brighter side of things, and I always consider plants are wonderful. But we have branched out into the animal side of things as well. So thank you very much. Thanks for patiently listening to me, even interruptions, and happy to take any questions if
Thank
the
you,
time.
madam. A very nice.
Director Mitchell, for your excellent presentation.
Thank you.
Hello.
You can go to the next one.
Hello. So, in the case of mixed infection of viruses, how does double-stranded RNA will be specific?
It depends on your virus. So if you are targeting, let's say your plant is infected with 2 or 3 different viruses, if that's what your question is.
Yeah.
Then you will have to have so that's what we are doing. We are looking at a crop like tomato. Okay, it gets CMV, it gets A, B, C, D virus. We can make a cocktail of dsRNA for those viruses. And also in our design, we look at the range of isolates. So we can target a conserved region among the various isolates of a particular virus. So the spray is really specific to that virus, but we can have a design which can cover multiple viruses.
Okay, thank you. So as in the case of the immunity, because pathogen might be also trying to overcome the RNA interaction. So is there any work related to that? How does a virus overcome the double-stranded RNA spray?
Yeah, so viruses can overcome RNAi. They code for what are called suppressors of silencing. So viruses code for suppressors. So one of the strategy that we are using is we are actually targeting the suppressors themselves. So, in the case, especially in the case of virus, you can target either the code protein gene, but you can also have a construct which has code protein as well as the suppressor of silencing. So that is one of the ways where we can approach. And also, sorry, maybe in order to answer your question, I should have mentioned that this technology is not curative for viruses. If a virus is already present in the plant system, it would have made enough copies for this small amount of dsRNA that we are providing to be effective. So it is more a preventative strategy for viruses. For insect pests and fungi, it is a different equation. But for viruses, it protects the plants before the virus comes.
Okay, thank you, Doctor. Thank you, Doctor.
Madam, what if a trait is governed by multiple genes?
So in order to answer your question, we are not targeting any endogenous genes. Okay, so we are not looking at changing the host in any way. Host is the same host. What we are trying to do is targeting the pest. So in the pest, we can pick up multiple genes if we think that targeting multiple genes will kill the pest or kill the fungus. But most of the time with viruses or with fungi, even if we pick up a single critical gene, it is fine.
Okay.
Thank you, madam. Now I request the next speaker, Dr. Nakiran. He is graduated from Tamil Nadu Agriculture University and postdoc from Canada. He has 25 years experience in biological control, guided several PhD students, worked on AMP genes of bacillus, and he also established biocontrol labs at different countries and published 140 research papers. Now, Dr. Nakiran, please go ahead with your presentation.
Yes. It takes some time for opening this PowerPoint.
Please go ahead with your introduction. Other things, they will make the slides.
Yeah. Okay. So can you see the slides now? Okay. So at the outset, I'd like to thank the organizers, Dr. Devappa, and the chairman and co-chairman for giving me this opportunity to share my views and the research which we have carried out on our journey on MAM-triggered immunity mediated by bacterial antagonists, which might be the weapon for the plant disease management. So according to the natural scenario for agriculture, we are aiming towards the promotion of organic agriculture and double the income of the farmer. So this is the...
Your screen show. Share the screen.
Yeah.
Slides are not seen, sir.
Okay. Thank you. Yes. Stop. Share. Can you, I think.
Sir, have you clicked on the share button, sir?
Yeah, click on the share button, and here it comes as you are sharing this. You are sharing start.
Yes.
Comment comes as you are sharing start.
Yes,
Probably
sir.
the file size may be bigger, so that might be the reason.
Go ahead, sir.
Okay. Okay, because I think this, can you please see here the slides are still the slides are not visible.
Slides are not visible, sir.
I don't know what is the problem. Because still the sharing on is still it is found to be rotating. It's not.
Maybe some network issue. It will take maybe 1-2 minutes, sir.
Anyhow, let me try. Time is limited, so I think I can. Can I proceed?
Yes, sir.
Your presentation slides, they will come later. Dr. Nakiran, please go ahead.
Okay. So regarding this, the management related to this, so the people everywhere working towards microbiome. So the diversity of the microbiome may be of two different types, which can be of endomicrobiome and the rhizomicrobiome. So in the case of endomicrobiome and diverse microbiome, in the present day, if you see the banning of insecticide order 2020, so more than 27 insecticides has been banned. And based on that, if you see, most of the fungicides which has been commercially used has been totally banned now. So now, if you see the products, biopesticide, which is available for the management of the plant diseases under the CAB registration, the products that are found to be available is only trichoderma, second one is pseudomonas, and third one is bacillus. But if the situation continues, so what is the next alternate that we have in our hand? So what is that we can think forward for going for the next generation, and what are the other new organisms that we can use? So in this concept, we went our journey. So we were traveling along with that of the endophytes, where we are trying to increase the in-plant resistance of the plant against the viral diseases, bacterial diseases, and fungal diseases. And based on our journey of the endophytes, so we explored the biodiversity of the endomicrobiome, which is found to be present in the Nilgiri biosphere. So based on the biodiversity which is found to be available as an endomicrobiome in the Nilgiri biosphere, we came across the various diverse nature of different species of bacillus, where Bacillus subtilis, amyloliquefaciens, and Bacillus pumilus, Bacillus cereus, Bacillus licheniformis, Bacillus methylotrophicus, and Ochrobacterium species were found to be identified as different types of the bacterial endomicrobiome associated with that of the Nilgiri biosphere. At the same time, the next hypothesis that we came into the frame is that whether the endophytic bacterial population of Fusarium marxosporum cubensi resistant banana cultivar play a role in antifungal activity or not. Because everywhere there's a huge cry stating that FOC race 4 has come, or race 1, race 2, race 3, race 4. So the continuous evolution process is going on. At the same time, if you see, there are certain genotypes where they are found to be totally free from the infection of Fusarium. So what's the reason behind that? So the resistant genotypes are found to be free from the infection of the FOC race infection. So where we came to a hypothesis that does the endophytic bacterial population of FOC resistant banana cultivar, they play a role in antifungal activity or not. So this was the core hypothesis that was framed. And based on that hypothesis, we have gone for the isolation of the bacterial endophyte, both from that of the resistant genotype and from that of the susceptible genotype, pertaining to that of the rhizome, pseudostem, petiole, and leaf. So to our surprise, what we saw was there was a very huge diversity in the phylogeny of the bacterial endophytes that was found to be associated with the resistant and susceptible genotype. In the case of the resistant genotype like OIKM5, if you see the phylogeny of that plant, a totally different type of endomicrobiome was found to be noticed. Out of which, if is the most of the endomicrobiome from the resistant genotype, we are trying to document those things to assess its antifungal activity against Fusarium marxosporum cubensi. So, based on our investigation, the most of the isolates pertaining to Pseudomonas aeruginosa, Ochrobacterium intermedium, Brachybacterium paraconglomeratum, Ochrobacterium tritici, Brucella melitatis, Achromobacter xyloxidans. So these organisms were found to be identified as an pathogen, that is antibiotic, which is having response for the suppression of the Fusarium marxosporum cubensi both under field and lab condition. Then the question that was raised in our case was, how does these endophytes are found to be differing in their mode of action compared to that of the same type of organisms that is found to be existing in that of the susceptible genotype? So,
sir, my PPTs are not visible, sir. PPT is not visible.
I don't know how to share, because I have clicked the share content also, share web browser, otherwise.
Sir, please send the PPT to the organizer, sir. Then it will be very good.
No, I think for right now, it will take much of time, I think, because it is around 30 MB, I think, size.
Otherwise, you make it 1 or 2 PPTs, 2 different, so that it will be good. Because you are...
Yeah, because I also feel the difficulty here, and because simply talking and you cannot see the slides means totally it is leading to.
Yeah, that's sir.
Yeah, I can understand all. Not synchronized. They say that it is not getting synchronized.
Sir, if the size is more, no, sir, you make it two different, sir, 2 different PPTs. You can share 2 twice.
Okay.
That option you can try, sir.
Okay, what I can do is, I think otherwise some of the next speaker can talk, And
Yes, sir.
during the night time, I can do that. Sorry for the inconvenience.
No problem,
Because,
sir.
No. Dr. Sheshakiran, can you mute yourself?
Yes, sir. Yes, madam.
Sir Nakiran, sir,
yeah.
you can email, if possible, if you can send us the PPT, we can open it here after the next speaker.
Okay.
Or if possible, you can make it into a smaller PPTs with few slides.
Okay. Then, okay, please request the next speaker, please.
Okay.
Sir.
Thank you.
May I request Dr. Rao? MS Rao is ready? Hello, Dr. Rao? Dr. Rao? Dr. MS Rao?
Sir Rao, sir?
Sir, I am ready, sir. I am ready.
Devappa?
I am ready, sir.
Rao, MS Rao?
I am ready. I am ready.
Okay, very good.
Once Nakiran, sir, close your one, sir, PPT.
No, now can you see the PowerPoint? My PowerPoint.
Yeah, we can see.
Yes, we can see reconfirmation of antifungal activity.
Yeah. Can I proceed?
Yes, sir.
Please go ahead. Please go ahead.
Thank you, sir. Thank you.
Please go
So
ahead.
here, we went for the reconfirmation of the antimicrobial substances produced by these different bacteria. So based on the confirmation of this antimicrobial activity, so the compounds were found to have in the zone of inhibition. So generally, we go for assessing what is the inhibition zone alone. So here we have extracted what is the compound that is going to be present here. So these antifungal substances were isolated and they were reconfirmed. They were identified as 3-oxymanolactone, then phosphothiolic acid, glucuronic acid, clindamycin, carbonic acid, and valeric acid. So based on that, if you see here, based on the ditrophic interaction between a pathogen and a fungal, that is, and a bacterial antagonist, the array of the spectrum of the non-volatile organic compound production was found to be totally different. This red color indicate in the case of the pathogen alone. So the compound profile that has been produced as a secondary metabolite during the ditrophic interaction is found to be totally different, which are found to be responsible for the suppression of the pathogen which is found to be associated. So these green color area indicate the compounds that are found to be totally different compared to that of the healthy control and inoculated control samples. So next, the question that we raised here was, does the bacterial endophytes from susceptible cultivar also inhibit Fusarium? So, but here, when you go for assessing the endophytes of the susceptible genotype, the total phylogeny of the susceptible genotype, the bacterial endophytes are found to be totally different. If we see the graphical representation, the susceptible genotype, namely NAPU1, consists of 3 different types of bacteria, namely proteobacteria, alpha, gamma, and firmicutes. Whereas in the case of the resistant genotype, it consists of 5 different group of bacteria: alpha proteobacteria, gamma proteobacteria, firmicutes, next actinobacteria, and bacterio. So these are the different types. So based on this, we took this forward for biohardening of the susceptible genotype with that of the bacteria that has been isolated from the resistant genotypes. So this is after the biohardening, the plant growth, vigor index, and everything was found to be more when compared to that of the control. And based on that, when we analyze the root architecture of the treated plants with that of Brachybacterium, Paraconglomeratum, Myroides, Achromobacter, and all those things, so the root architecture was found to be totally altered when compared to that of the untreated control. So here, the root architecture is found to be, so this is the close-up view of the root architecture, which has clearly indicate that the root proliferation is found to be increased because of the use of the endophytes from resistant genotype. So this is the case where the disease incidence in the case of the control and in the case of Brachybacterium treated, and this is how the root architecture is getting altered. So the root architecture, the root depth and penetration, lateral roots, all are getting increased. Whereas in the case of Fusarium inoculated control, the root architecture is found to be totally collapsed over here, and the number of roots and the biomass of the root, the distribution pattern is found to be altered. So the next we went for seeing whether the bacteria is getting associated with that other root region or not. So the scanning electron microscopic photograph was taken using Brachybacterium. When you are biohardening with Brachybacterium, the root region was examined under the scanning electron microscope where we were able to see the presence of Brachybacterium under the electron microscope. Not only on the root region, the bacteria moved in an endophytic manner, and from the pseudostem region also we were able to document the presence of the particular bacteria, and from the leaf portion also. So bacteria was found to be associated. So this is the case with the next isolate, namely Bacillus amelia. So this is the bacterial biofilm formation, which you can see under scanning electron microscope. When you are observing the root under the scanning electron microscope, you can see the very huge formation of the biofilm formation. So when this condition is found to be persisting, just kindly imagine what might be the fate of Fusarium to gain entry into the system. So the Fusarium conidia is also found to be suppressed in this case. And apart from that, in the pseudostem also we have documented. And this is the tritrophic interaction where different types of the chemicals, what are the different type of chemicals that has been induced during the interaction of this beneficial microflora. So when this is, so it is resulting in linking with the different types of the pathway where TCS cycle is connected, citric acid cycle, glycolysis, triterpenoid pathways, fatty acid biosynthesis, beta oxidation pathway, and alpha linoleic acid metabolism. So these are the different types. This red colored one indicate these are the different types of the compounds which has been produced, and they are found to be associated in interaction with that of the plant defense mechanism, which results in suppression of the disease associated with that. So is it a case associated only with that of the fungal pathogen? Next, we went for investigation how these particular bacterial endophytes play a vital role in suppressing the viral diseases. For example, we work on ELAR virus. So in the case of the ELAR virus, the bacterial endophytes was isolated from the healthy plant and the infected plant. So if we analyze the red color, at the bottom, if you see this particular graph, the rose color one indicate the bacterial community which has been isolated from that of the infected one. Whereas the green color indicate the bacterial flora which has been isolated from that of the healthy one. So this is the documentation of the rhizosphere bacterial community and endophytic bacterial community which is found to be associated with the healthy TSV infected plant and that of the healthy plant. So this one, if you see, so the bacteria which has been already isolated and documented. So these are the antagonistic bacteria with the known antimicrobial peptide profile, which we have published in the Frontiers in Microbiology, this particular paper, where Bacillus amelia, subtilis, megatherium, primulus, lacaniformis, methylotrophicus, and their tried for its antiviral activity, where the plant was challenged. The localization host was challenged through pre-inoculation spray, simultaneous inoculation, and post-inoculation. So when you go for pre-inoculation and simultaneous and post-inoculation with respect to Bacillus amelia in the localization host, the symptom expression was found to be totally arrested. There was no symptom expression, whereas rest of the bacteria was not found to be equally performing well according to the other one. So among the various bacteria that we have tested, the endophyte Bacillus amelia and Bacillus licheniformis were found to have 100% reduction in the lesion of the ELAR virus or the tobacco streak virus in cotton, whereas you can see the innumerable number of lesions in the case of the inoculated control, which is an evidence which is found to be present in front of us, stating that it is found to be having a role in suppression of the virus lesion. The number of lesions, if you take here, so the number of lesions is found to be reduced in the case of Bacillus licheniformis and Bacillus amelia. So the total number of lesions has found to be totally reduced in irrespective of different types of inoculation, both in pre, simultaneous, and post-inoculation. Next, the percent tobacco streak virus incidence was also found to be comparatively reduced in the case of the bacteria which was used for challenging, namely Bacillus amelia. Then if I take the plant height, number of flowers, number of bolts, and seed cotton yield, all these plant growth promoting parameters was also found to be increased because of the application of this bacterial endophyte, namely Bacillus amelia. And then we want to calibrate what was the virus titer which is found to be associated with the plant after having the infection. So here, if you see, in the case of Bacillus amelia treated one, and in the case of licheniformis, the virus titer was found to be very, very reduced compared to that of the inoculated control, which has a 2.5-fold absorbance value, whereas in the case of the bacteria challenge, endophyte challenge 1, the virus titer was less than 0.5, which indicate that the virus is found to be, the endophyte is found to be working in this particular system. So this is the field trial which we are done, and this is the summary of this particular one, where how does this particular endophyte is found to be acting in the suppression of the disease associated with that of the tobacco streak virus. Then what is the reason behind the suppression? What are the treasure in the genome of the VB7? So that was the curiosity for which we went for whole genome sequencing of this particular one. So based on the whole genome sequencing, we came to a conclusion that baseless amyl liquefations was found to be very closely similar to that of the baseless velizensis, and amyl liquefations was identified as baseless velizensis, which consists of 14 secondary metabolite cluster. So the non-self molecule which is produced from 14 secondary metabolite clusters must be responsible for the suppression of the virus replication in the plant system, and it consists of the clusters which are responsible for the synthesis of surfactant, bacillibactin, bacillizin, difficillin, penicillin, and so on. So next, we want to understand how does this particular one behaves for the other virus, namely the TOSPO virus. How does bacillus interact? So for which, how does MAM-triggered immunity is going to be working here? So for which, what we did is we cloned the gene, elongation factor gene, and iturin gene. So the iturin and elongation factor gene has been cloned to the top the local lesion host, and where the number of lesions was found to be reduced in the case of the pre-inoculation spray. So these are the symptomatic expressions where the metabolites of bacillus species, they reduce the symptom in the case of the local lesion host for the tomatoes, for the GBNB, where in the case of bacillus the amyl liquefaction simultaneous inoculation, the lesions is found to be very much reduced here. And next in the case, so this is the one. So it is found to be playing a vital role in reduction of the for the development of understanding the mode of mechanism of action. So the constructs was developed using the iturin gene and elongation factor to understand what is the MAM-triggered immunity is found to be taking place. So these are the MAM-triggered immunity. So the non-self molecules associated with this particular bacteria, namely the bacillus velizensis was flagellin, elongation factor, peptidoglycan, lipopolysaccharides, phenylalanine, cell wall beta-glucan, and cell wall chitin. So here, when we do this, so we have constructed the clones. Where is there any regulation of the different gene by the clones, or if it is so, what is the impact on suppression of GBA? The rhizosphere zone of the tomato PKM1 seedlings, and after that, the plants was challenge inoculated with that of the GBNV virus, and after that, the different gene expressions was found to be analyzed, where we were interested in understanding the different genes, namely mitogen activated protein kinase, NPR1 gene, then Ricky transcription factor, and PR1 gene. So when this was analyzed, the transcripts was analyzed in the case of the inoculation, where irrespective of the different treatment, that is 0 hour after inoculation, 24 hours after inoculation, 48 and 72 hours after inoculation, with that of the groundnut bud necrosis virus, what is the level of expression of the transcript of mitogen activated protein kinase, which is a key gene which is responsible. So that was found to be increased in the case of bacillus, and in the case of elongation factor, and in the case of iturin. So in the case of the NPR1 gene also, the transcript level was found to be more, and in the case of the other gene, namely here, the PR1 gene transcript level was also found to be increased. Next, the Ricky genes, the transcription factor was also increased because of the application onto the thing. So when these transcripts are found to be getting increased in the plant system, so what is the symptom severity grade that we have assessed? So the symptom severity grade was found to be very much reduced in the case of MAM genes, that is the genes agrobacterium with flagellin, agrobacterium construct with elongation factor, and bacteria as a whole as such. So the lesions were found to be very much reduced. So the absorption value of the coat protein was also analyzed, where the coat protein was found to be very much reduced in the case of the plants that has been challenged with the agro-infectious clones. And here the transcript copy number, the coat protein copy number, was also assessed through real-time PCR, where the number of copy number in the case of inoculated control reached up to 10 power 8 after 9 days after inoculation. Whereas in the case of the treated one, the inoculation, that is the virus titer, was found to be more than 200-fold reduction in virus titer was observed. So in summary, if you want to say, the MAP-associated molecular pattern was perceived. After the perception, the mitogen-activated protein kinase is getting activated. So the oligomer of NPR1 is converted to the monomer, and this particular NPR1 reaches the nucleus region, nucleoplasm, and there it goes and triggers the transcription factor, namely the Vicky transcription factor. After increasing, it increases the PR1 gene, the transcripts gets increased, and because of the increase of this, the virus lesion of the virus titer and the lesion is getting reduced, and because of that, the symptom severity under the field condition is found to be reduced. And this is an ultimate diagrammatic representation of the graphical abstract of the mode of action of the MAP molecules of elongation factor and deuterium. So how they interact and how at the downstream end, what are the TGA and what are the other transcription factors that has been induced in the plant system to overcome the infection of the groundnut bud necrosis virus in tomato. So in short, the researchable issue that is placed in front of us is, so we can go for the exploring the endomicrobiome and development of suitable techniques, and boosting the immunity of the plant growth for harnessing the synergistic action can be better understood based on this. So finally, I would like to acknowledge my lab team members. So these are the team members of our lab, along with Dr. V.G. Malati and all our crews who are responsible for the development of this particular work. And once again, I thank the organizers for giving me this opportunity to share our research experience on MAP-triggered immunity for the management of both the fungal and viral diseases. Thank you.
Thank you, Dr. Nakiran. Very nice presentation. And you have also kept up timing in spite of some difficulties. Very good. Thank you very much. Audience,
thank you, sir.
One or two good questions, please. Hello. Anybody have any questions, clarifications?
So the questions, okay.
Sir Nakiran, sir?
Yes, madam.
Sir, what would be the response of the other bacteria or fungi which are there in the phylloplane or the rhizosphere? How would it react for a new ecophyte which you're going to introduce?
Okay, so here in the case of the introduction, we are going to deliver this on the plant at the rate of 10 to the power 8 colony forming units. Okay, so when we apply this, the MAP molecules which is found to be present, so there are more than 14 to 15 non-self molecule potential molecules which goes and trigger the MAP triggered immune response in the plant system. So suppose if there are some other microorganism associated with that, and they can also have a synergistic interaction for triggering the immune response in the plant system. If it is of pathogenic origin, then there can be a chances for, suppose let us talk about a virus means, then if the virus inoculum load is going to be more. Under that circumstances, then the chances for the reduction in immune response may be there. So the presentations in the case, in most of the cases, pre-inoculation is found to be effective, which indicate that prior to the infection of the virus, if these MAP molecules gains response with that of the pattern recognition receptor, then the immune response can be triggered, and which can be highly effective for the management of the virus.
Sir, one more small question is there, if you permit. Sir, I'm very happy to see your work on banana, especially by hardening in NAP1. I want to know whether you confirmed the disease resistance, whatever you observed in the nursery level. That you tested in the field for the resistance.
Now we have completed one field trial, and there's another field trial is also going on. So we have taken the biohardened plants, it has been taken to the field condition, and based on that, our experience, a bacteria, namely the Bacillus velicensis and Achromobacter xyloxidans. So these two bacteria are found to be performing very well in the suppression of Fusarium pubensis infection under the field condition. So this trial, we have completed the trial, and yet we have to. So after publication, we will be presenting all those things.
Okay, sir. Rastal is a main variety.
Your voice is muted. The what?
Yes, sir. So Rustali is important variety, which is more susceptible to fusarium. Have you included Rustali also in your study?
Because we can include, there is no issue in including that. If someone is ready to collaborate with us, we can have an exchange of the technology and we can start working on that, because it is a PhD student work. So in that case, the susceptibility, that is the infection, is around 30% under the farmer's field condition in that is NAIPU 1. The infection. Suppose if, because since it is a student work, I don't think that extending Rustali may be possible. I think in subsequently, if there is some grants, if we are getting, then we can extend for Rustali also.
Thank you. Thank you.
Thank you, Dr. Nakiran. Very nice
Thank you,
presentation. Now, I am very happy to introduce the next speaker, Dr. Ramesh Rao, whom I know for more than 40 years. He was the head of the principal scientist and head of the division of entomology and nematology at IIHR. He has granted several international patents, national patent, one national patent, and has developed different technologies and transferred to more than 525. And in industry licenses in India was a world record. Very outstanding contribution. He has received several awards and honors, and he was a fellow of several scientific societies. And one important contribution he has made is, through the technologies developed by him, about 1,000 crores of biopesticides are produced in India every year. Congratulations, Dr. Rao.
Thank
now
you.
I invite you to make a presentation.
Right. Thank you, sir. Thank you. So I would like to thank Professor Devappa and Dr. Krishnappa and all others. So I'll start with my presentation. So yeah, so straight away I'll just go ahead with the the whole situation when we talk about sustainable agriculture or organic farming. The farmer understands actually only the health of the crop and health of the soil. And when we think about actually being a pathologist, we only think about pathogenic bacteria, fungi, and being a nematologist think about nematodes, and then the entomologists think about insects. But the truth is that actually there are interactions. So we have to address all these things and all. So I just want to give some examples. In the case of Goa, so you have Fusarium and root knot nematode, and the problem is extremely serious. It starts from the nursery stage, and then the whole plant gets collapsed. The farmers having a serious problem throughout the country. And if you see this, actually, this is all the pictures which I'm showing it. Actually, the situation is very serious. We say it is a Goa wilt. And Goa wilt, actually, it is not because of only, see, when we start thinking about only from the pathological point of view, it doesn't serve the purpose because it has a complex. So we'll have to understand, even in case of pomegranate, again, it is a complex. It's a ceratocystis and root knot. Yeah. And this is okra, you have Fusarium and root knot nematode. And in case of tomato, you have actually Fusarium, Ralstonia, and actually the nematodes. And in case of carrot, actually you have Erwinia. In case of gerbera, you have hello, somebody has to mute, please. Somebody has to mute, please. So in case of gerbera, there is again Phytophthora and root knot nematode. And in case of capsicum, we have Ralstonia and root knot nematode. Same is the case with the tomato. It has three pathogens actually. And carnations, again, it is a complex. And the gerbera, again, it is a complex. So you take actually the polyhouses, this problem is very rampant and it's very serious. Nematodes are also very, very serious. So this is how actually the whole situation. And so then, see, this is how the nurseries are, in India, precisely, whatever, we say actually, they carry at least 30 to 40% of the seedlings carry the pathogens from the very beginning. The farmer takes it, and in his soil, actually, the pathogens coming from the nursery beds, and then you have the wilt problems, whether it is a bacterial wilt or it's a fungal wilt. And this is all the scenario I just want to share actually. So when we talk about the biopesticides, from the larger perspective, actually, we'll have to think about the soil health and the plant health. And when these two, soil health and plant health is taken care, animal health and human health is taken care actually. So this is how actually nowadays, even Dr. Nakiran also spoke about microbiome. It's not a, just one organism is going to solve all the problems. We'll have to have the microbiome approach. So the problems are actually whether it is because of the bacteria or fungi or nematodes or insect pests. So the microbiome approach, rhizomicrobiome or the endomicrobiome, this is how actually the manipulation. So the whole biopesticides will fit in this particular area. Manipulation of the microbiome and manipulation of the the whole soil ecosystem for the benefit of the soil health and also the plant health. This is the concept. This is the concept which I envisaged actually as a nematologist. I was not successful with the bio-nematicides. Then I had to look into the aspects of pathological aspects actually. Then I had become a pathologist, learned from the pathologists and learned from the students of have done the pathology. And then ultimately we started integrating actually the whole plant production, and I became an entomologist, being in entomology department of IIHR. So we started actually, doing it actually in a holistic way. This is what I advise all the people actually. You may be an expert in one particular area, but when it comes to the question of delivery and application, farmer point of view, sustainable agriculture, it has to be a holistic approach. From the point of students, I just want to show that actually, when you want to do any research, you'll have to be very sure that what organism you are, isolating, and see that your isolate is actually sure that what you are doing it, and have the molecular and diagnostics and all. Utilize all these technologies and see that actually what you are doing, actually what you are handling is very clear. And this is how actually you'll have to start your work. Actually, you'll have to start with basically with identification. So when you have an efficient strain, actually, you should see that it is characterized completely, and understand actually it's a genetic basis, and see that actually all this process, I'm just rushing through because I have so much to share. Just only from the point of the students, see that you follow all these steps actually. Molecular characterization, biochemical characterization, morphological characterization comes in the beginning, and efficacy evaluation. And then if there is a time and there are facilities, think about the molecular basis actually. See, we started actually, we started telling trichoderma works against nematodes. Then pathologists and the whole horticulturists and all the agriculture people were asking actually what is the basis? Then we said actually there is a nematicidal gene present in that. So this is about the nematicidal gene actually, which we worked out. And we have gone into the depth actually completely, showing that actually a genetic basis. So this is a gene expression actually. So this particular gene expression is ultimately with this we could show that actually the plant how it responds and what is the gene expression and how the nematodes are getting affected. So we wanted to show that actually how the nematode X actually are getting affected. See, the nematode X are getting affected here. So this is how, if you can find the basis, whenever, you go for the DBT or whenever you go for the DST funding, do you have actually, proof? They say actually, proof of concept. So whether you have an organism which is very effective, whether it is, proven one, whether it is molecularly characterized, do you have the, basis actually, all this, people ask actually. I'm sharing this as, all these things from the point of the students. So, or the young researchers, if they want to go ahead, actually, they want to work actually on the biopesticides. And Dr. Krishnappa was telling actually, my work led to the, dissemination of the technology among the industries, and industries are producing, Trichoderma, Pseudomonas, Bacillomyces, and Poconia in a big way. This has happened actually because of the groundwork that we did actually with all this, data actually. So, it is not that you are a pathologist or an entomologist or a nematologist. No, you should become an agricultural scientist thinking from the point of the farmer. So farmer faces a different scenario. The soil health, if you see, it's a totally different scenario, and the plant health, if you see, the whole ecosystem actually, you have to have that approach. So you have to see where your biopesticide, where your candidate, which you have been working probably for 5 years, 10 years, where you can fit in. So that is what actually I show. So we started seeing actually volatiles, do they have any effect actually on this, from the Pseudomonas montelli and from the Bacillus pumilus, what is the effect of the volatiles and how it performs in the dual culture, and all this, all this actually is the basis. All this is a basis actually when you want to see what happens to conidia, what happens to the, nematode eggs actually, and how the nematode eggs get infected. All this basic work you have to have. Whether this same Bacillus pumilus, how it works against the bacteria, pathogenic bacteria, and against the fungi. So you have to generate actually, you may be a pathologist, but collaborate with the nematologist. Nematologists collaborate with the, other, pathologists, virologists, and the, bacteriologists. This is how actually we'll have to, try to solve the problems, having understood actually that we have burning problems, but the burning problem is not only because of the fungal pathogen or bacterial pathogen or nematode alone. It is a complex. Thrips alone actually causes, but then this when we take care of the soil health, the thrips population of the plant comes down. So the viral management happens. So this is how actually effect of nematodes we have done, and on the X actually it has happened. Then we did the GCMS studies, and then the volatiles and how it is done, and split root technique. All this is to say the basis that we have a candidate which is very efficient, which works very well, and it is proven, and it's a molecular basis actually is completely worked out. Then when you go, whatever you do it actually, you will have to experiment in the field conditions, not only in your university conditions or in your experimental conditions. You will have to think about going ahead with the farmers. Sick plots, you will have to identify, take the help of the farmers. Farmers need help actually. You will have to see that actually your candidate performs well. So this is what actually is required. This is what as it is. When you do this experimentation, you will have to generate a lot of data, so that you can publish it. You can publish it. This is about the Bacillus annulifications. So the data, when you want to do it actually these days, it's a very highly challenging. So when you want to go become a professor, then you go for interview, or you got to go to the director. They say high impact publications. High impact publications can happen only when you have a sufficient data. So it's only to keep young scientists, young students. I'm sharing all this actually. This is all the data. It's a publishable. See, this is all the data actually. This is required by the editors, and then it becomes a sound publication. I'm showing all this information actually rushing through, just because to prove that actually what we have done is a base, has got a scientific basis, and it is a sound, and it is a publishable, and it's a patentable, and it works well in the farmers field conditions. This is what actually I just wanted to tell about the this is how a biopesticide research is not like actually you take a chemical and screen it is good, and you take you take one rootstock and you screen it is good. This is not the thing. Biopesticide research has to be holistic. You have to understand the plant as it is, and you have to understand the soil's ecosystem, work with the soil scientists, work with the agronomists. So this is how actually you'll have to work with the farmers. When you see the farmers are losing heavily. So if I as a nematologist, when I go and say, okay, you apply Pathology, no, I'm not doing justice, because he doesn't get actually any relief in this, because there is a bacteria, there is a fungus. So we have to think about the holistic approach. So the holistic approach, whenever you think about the biopesticides, releasing biopesticides through the drip irrigation or anything doesn't work. There are billions of the pathogens in the soil. You need billions of the propagules of the bioagents. So for which you will have to think about the organic matter. So the organic matter actually, in India, we have manure application and the vermicompost application, neem cake application. They have to be enriched actually. They have to be enriched, and we have to think about application of all these bioagents together along with this actually. So we start with the soil, from the beginning actually, how to treat the soil. Treat the soil with the farmyard manure, with enriched with all the biopesticides, and you can have a biofertilizers. people, started speaking actually, what about interactions? What happens to my candidate? Nothing happens actually. Think about the forest ecosystem. There is Azospirillum, there is a Bacillus, there is a Bacillomyces, there is a Nitrobacter, everything is there. This is what is called sustainable agriculture. There are competitions. There is a, registrary competency, but works out, when you go to the organic matter, everything works out well actually in the soil ecosystem, like the forest ecosystem, which is working very well, where you don't actually interfere or manipulate. But here, if you do that, actually, you will go closer to forest ecosystem and you can reduce the chemical use actually tremendously. This is what is a sustainable agriculture. Organic farmers, they say only FIM and organic matter. And the pathologists will say only, through the drip actually apply the, some particular bioagents. No, this is not the way. Soil scientists speak about the fertilizers, and Syngenta Bayer speaks about the chemicals. No, this is not the research. This is not the science that India wants. Farmer wants. When we talk about the doubling of the farmer's income, he has to have first of all a healthy soil. If he doesn't have a healthy soil, actually, then the sucking pest management is highly difficult. When you have a healthy soil, healthy roots will be there, and the healthy plant will be there, healthy plant canopy will be there, and induced specific resistance works actually, and then the robust ecosystem plant you are producing, immune plants you are producing, and you can see the this all happened with the 90% of this actually. This gentleman actually has used biopesticides along with the every kind of drenching, along with the neem cake suspensions, and along with the farmyard manure, and he had to use only 10% of the chemicals, which is very difficult in the polyhouses. Polyhouses, people are dumping actually 90% of the chemicals, and they say 10%, 20% they use biopesticides. They don't work. This is not right actually. You should know actually how it works. We should understand the biopesticide actually how it has to be introduced. The delivery system is very important. The problems are very complex. I'm showing you actually this is all the very things. Farmer doesn't even understand there is a pathogen inside. He only thinks it is a soil problem. It's a nutrient deficiency. No, you have issues like this. Crores of rupees people are wasting. People are dumping the chemicals. FAO has come out saying that India is going to contribute tremendously next 2 decades actually, because of the soil health problems, and where in polyhouses, protected conditions, people are dumping the chemicals. Yes, there is a FAO report is available, but then it's not a big, difficulty. So you should think about, using the biopesticides actually on the beds from the beginning, from the, preparation, then you get the healthy crop. So like this, we have so many success stories, and so many NABARD people used to come and they used to bring the, farmers, and we used to show to the farmers, we used to show to journalists, we used to tell the journalists actually, bring the awareness. And I have been sharing among the pathologists and entomologists and the horticulturists, agronomists, saying that actually don't talk about FIM only. Talk about a holistic organic farming, a bioorganic farming. Pathology speaks only about biopesticides. No, let him talk about the soil ecosystem. Let him talk about the enrichment. Let him talk about the soil delivery system. Let us be people who are interested in the farmers' problems. Let us not say that actually I have a DNA fingerprint and I have good pesticide bioagent. It's very effective. No, this is not science. You have to become a sustainable agriculturist or a horticulturist. Then only you will be able to help the farmers. A holistic approach demands all this actually. So these are all the so problems are very complex, and when we don't take actually soil health into consideration. So simple biopesticide will not solve the problem. Again and again, I am telling actually you will have to introduce along with organic matter. The organic matter only enriches actually the millions of the Trichoderma propagans becomes billions actually when you go into the along with the organic matter. This is how the success of the soil health and the plant health. And these plants are highly robust, resistant to the thrips and the mites. And the red mites is such a serious problem, but they didn't have to spray more of chemicals. And this is how actually, the neem cake can be enriched, organic matter can be enriched actually. So after enrichment and the biopesticides develops tremendously. The soil is healthy, pulsating with the millions of billions of actually bioagents here. Before the plants are sown, a farmer spends around 2 to 3 lakhs up to 5 lakhs for 1 acre for gerbera and carnations. Out of 5 lakhs actually he loses almost around 2 lakhs because 30 to 40% there is a mortality, and over and above that actually he spends drenching chemicals, which is absolutely ridiculous. think about the soil, how to prepare. When the mother is conceived, mother is prepared actually by 3rd month, to have a completely, iron, zinc and calcium, everything is given. Same way, then the baby, is healthy. Here, the baby which we are planting cannot be healthy. Within 10 days, actually, the farmer is, losing a lot of money. The soil is becoming sick again because he is using the chemicals. So this is how actually you will have to see that the soils are, fully enriched. Every organic matter actually should not be, 1 gram of organic matter actually should not be used without adding biopesticides. This is a slogan I propagate, and I'm a sustainable agriculture campaigner for sustainable agriculture in India. So I always say that actually whenever you use even neem cake, don't use the neem cake as an organic amendment. It's a precious, it's a neem gold. So enrich with the biopesticides. Then you see the success. You have a growth promotion because of the induced systemic resistance and because of PGR effect, and you have the increased yield and disease suppression. What farmer wants is this. Chemicals he doesn't use. And here the virus management happens. It's not because it has become a virus, resistant. It has become a tolerant. And then, the pests are not able to harbor. And this is how actually, the banana wilt actually, is a problem, Fusarium nematode. When in the presence of nematode, the robusta also becomes susceptible. But when you have, this was, in one particular plot where in 6 years, raccoons were taken actually without using the single gram of, carbendazim or any other chemical. And excellently, actually, they started using the biopesticides through all means. Through Jivamrita, through Panchakavya, they started using the biopesticides. And this is how the problems are rampant. The problems are rampant. The universities are not talking about, actually, they talk only about the, mixing of the various chemicals, mixing of the various bactericides and fungicides, but they don't talk about actually, IPM real, having, predators, parasites, biopesticides in a one systematic manner. You take the, any ETV program, anything and all. There are only IPM, they talk about chemicals, integration of chemicals. This is not the science. Please, think about, thinking about actually organic matter application and application of, all these things, sustainable crop production. And when sustainable crop production, start with the biopesticides, start with the biofertilizers, start with the micronutrients, start with the soil health. When the problem comes, actually, farmer, this season, okay, you do it, but next season, don't do it. This is what actually all the scientists, it's our responsibility. Agriculture department people will never know. They don't understand even actually application of even Trichoderma pseudomonas properly. So we will have to educate every one of them. We have to educate the farmers. So our role is very crucial. This is a gentleman actually YouTube, all these things what I have presented actually YouTube, they are available. All the success stories are covered by the Hindu newspaper and all the other in YouTube. They're all successful farmers who proclaimed and declared saying that actually they have used only 5 to 10% of the chemicals. So this is how actually, the success story. Then because of, I started working as a personal myself being a nematologist. Then I had to become a pathologist. Then I had to work with the Trichoderma. Then the industry came actually, I need your Trichoderma. So Pseudomonas and Trichoderma viridae. Then we started developing the data for the industry. So this is what is a commercialization. So when you have a good candidate, actually, you'll have to develop not only bioefficacy data, you will have to develop actually, toxicological data. You have to develop all the shelf life data. You will have to think about, approaching various industries. I have a wonderful candidate. I have tested in hundreds of fields, farmers' conditions. And then our director of research says that it's very good and I need funding. When you have a proper listing and all, there are people who will be going to fund. When you have funding, actually, you develop the data like this. So all this data from the CAB assay registration, and develop the data from the DPT and DST. And all this data ultimately was required for the industry. So when you work actually as a scientist, and when you don't work with the farmer, you don't understand actually, you don't get the success. You may have the patents, you may go abroad actually for presentation and all, but then you are not a real actual scientist, you will not have a self-actualization in your life. You may have hundreds of papers, but then actually, if you have worked actually hundreds of farmers, and if you have brought the relief, that's a real science. Then when the farmer wants actually a big Trichoderma pseudomonas is not available in the industry, then you have to think about the industry. Yeah, a small, biscuit actually it cannot come without a particular regulatory authority number licensing. So this license is required by the industry. So to produce actually, so the industry looks towards the scientists. So this is how actually the formulations, we started, developing the protocols, and we started working with the various, scientists 43 scientists from the All India coordinated research projects. Actually, all the coordinated projects, whether it is a fruit coordinated project, or vegetable coordinated project, or nematological coordinated projects, or the biocontrol research project, we got involved all of them. And everybody, said that actually reasonably your, biopesticides are good. And then, industry started coming. Then we started giving it to the industry. So this is how actually it has gone to the industry. So the patents, side by side, actually, this is an Indian patent, this is a US patent, and this is all the 7 international patents and the 1 international patent. Actually, I got it actually in my journey, and then we started working with the industry. We started helping the industry to go ahead, quality, production of the, pesticide, biopesticides, and all. We told them. Then all these things ultimately are helping the farmers in a big way and helping the nation in a big way. Quality issues are, yes, there are true. Actually, the quality issues are there, but then we are working actually with the industry. Go actually, the quality issues are going to be. So actually, the market potential is tremendous. So from IIHR, I have given the technology through almost all parts of the country, in all the parts of the country, in the union territories. Nobody can say, farmers cannot say, no, I don't have Pseudomonas, I don't have Bacillomyces, I don't have actually, Trichoderma. No, it is available everywhere. And then, you will have to think about, taking it, utilizing it in an effective way. So again, I just want to summarize in the interest of the young scientists. Working in applied aspects. If you want to be a basic scientist and you want to be applied scientist, no, you have to have both and science is required for the applied research. Then you will have to have actually a tremendous amount of persistence, perseverance, and patience to work with the farmer, industry, to work with the policy makers, to work to convince the authorities, and with passion as a mission, not as a job because you are getting paid. So biopesticide startups actually, if somebody wants to do it, and you will have to. Nature provides the complete data, and you will be able to help the farmers. So this is how actually I just want to say. I want to thank everyone, and this is my journey, and I want to share actually all these things in the interest of the sustainable agriculture, mainly the farmer. So please keep all these points in your view when you want to do research, and when you want to do work with the farmers, when you want to work with the regulatory authorities, and do this, and then absolutely success will run towards able to serve the. Thanks a lot. God bless you.
Thank you, Dr. Rao. Very nice presentation, and within a brief time, you have made all the important points very clear. Thank you very much. Audience, if you have any 1 or 2 questions, please. Anybody any clarifications or questions? Audience? No questions? Hello?
Sir.
Yeah.
Good afternoon, sir.
Good afternoon.
I'm Naglakshmi, plant pathology scientist, sir.
Okay.
This talcum formulation, some people are feeling that the active ingredient is very low, like that. Which formulation is best for mass multiplication, sir? Mass production?
Mass multiplication, even talk powder is also good actually. CABRC 6, you should have for the fungus, you should have actually 2 into 10 power of 6 CFU per 1 gram. For the bacteria, you should have 2 into 10 power of 8 CFU. There are industries with the talc powder itself, they are able to go up to 10 to the power of 9, 10 to the power of 10 also. It is possible. So don't discard actually talc powder as a useless media. It is a useful media. You will be able to achieve. Liquid formulations are also good, where you will be able to maintain more CFU. Don't worry about this apprehension. It is true. It is not. There is no truth in that. So you can have a very good formulation with the talc.
Okay. Thank you, sir.
Any other questions, please? Thank you, Dr. Rao. If there are no questions, thank you very much for a nice presentation.
Thanks a lot.
And now, as per the request of the organizers, we will not have any lunch break. Those who want to have, they can have individually. They can separately have the lunch. And we will continue with the session. And next speaker will be invited speaker, Dr. Jagirdar. If he is not there, Gurdat Hegde. Any one of you, please. Are you ready? Hello, Dr. Jagirdar. Dr. Jagirdar, please. Or Gurdat Hegde. Not there? Then we will go for the next speaker. Anybody who is present in oral presentations?
Dr. Rajput is there, sir.
Yeah, please. Rajput, please come in. Who is ready? Let us finish up. There are 6 presenters. Rajput?
Yes, sir.
Please go ahead. Please go ahead. You have 10 minutes time.
Okay, sir. Thank you.
8 minutes complete the presentation and 2 minutes let us have a discussion and questions.
Okay. Thank you. I'm Dr. Lakshman Singh Rajput, scientist from Indian Institute of Fire and Research. Newly I started working on swabbing enterprise for management of fungal and for management of major fungal disease of soil. Already, I don't want to give more introduction about endophytes. Already, you know, so many things about just now on various scientists. Just I want to say, major important disease which is facing by the soybean. From last 5 years, we are having 3 diseases like anthracnose, charcoal rot, and rhizotonia aerial blight. The fungal diseases which are giving the huge losses to soybean. Thus, in these pictures, we can see the entire plots are going. And this year, nearly 3 million hectares is facing the problem of both anthracnose and RAB on major soybean growing belt of Malwa region. Now, for that, manage to that, I isolated the different endophytes, bacterial and fungal endophytes. So I selected some wider and beyond the plant, JF55 and JF9560. They are widely grown plants. And after them, the varieties which are disease resistant, I think that 2098, 9752. That 9752 have the resistant towards various fungal diseases, as well as it's resistant towards both abiotic viruses also. And after that, I isolated from the different plant parts, from leaves, stem, roots, and everything. And to avoid the unnecessary bacteria, I use the things BVDA rather than nutrient agar. Nearly 136 isolates, 75 isolates I isolated, among the 80 fungal and 56 bacterial endophytes I isolated. What I found that if we can take that in resistant varieties, we are finding more endophytes, and in the case of leaf and stem regions, we are having more endophyte rather than the seed and root region. Most of endophytes are frequently, they are found in all the seeds, leaves, and everywhere. Directly, I went to that in vitro evaluation, and I found that few of the endophytes, I found that few endophytes only here, which is the in top region. Like in isolate number 3, which is actually the Aspergillus flavus, and isolate 41, which is the Aspergillus niger, and few various species of Fusarium. They perform very well, even though they can inhibit the mycelial growth up to 80%. They can inhibit in vitro conditions. Similarly, I isolated bacterial endophytes. I performed the first frame to all endophytes, and after the frame, I selected only 13 isolated for in vitro study. I selected from that. And again, I did in vitro analysis of all 13 isolates against Coledotrichum truncatum, Rhizotonia salani, and Mesopotamia pisciina, 2 stick plate assay. And what I found, a few isolates, they are nearly excellent. They also then that isolate number 5 and isolate number 111, they can inhibit more than 80%. They are penidecilous species. There are 2 different penidecilous species. And after that, I performed some biochemical assay to know their biotic potential. What I found that various enzyme, anti-DSO for various enzymes semi-quantitative methods, chitinase, proteinase, beta-1 gluconase, cellulase, and amylase. Some isolates they will produce the chitinase, most of them they can produce chitinase except 41 isolates, that is aspergillus niger. Other they can produce the various enzymes to more or less quantity. So this is the results of the various tests. Here we can see the high chitinase activity, high cellulase activity also we can able to see. And so like the amylases and proteases and beta-1 glucanases, the three enzymes are very slight produced. Interestingly, whatever isolates I have in bacterial endophytes, I have only one isolate, Bacillus cereus, it's able to produce chitinase. None other than can produce the chitinase. Then I performed the HCN test also, hydrogen cyanide. Most of endophytes they are not able to produce that hydrogen cyanide. Only the few, that one Fusarium and one Aspergillus niger and two Fusarium different species can able to produce that HCN. Interestingly, as we know that soybean is a oilseed crop and it requires phosphorus. So I tried to put phosphorus solubilization also. In that only one isolate of Fusarium species can able to produce. In bacterial endophytes, none of the bacterial endophytes is solubilized. Then I searched for some other characters also, like nitrogen utilization. Most of fungal isolates can able to utilize nitrogen very well. I go on the nitrogen free media and just I took the radial growth, the gradient growth. Most of isolates can able to growth in ammonium and nitrate both form they can able to uptake. And after that I performed the hydrophore assay, and I come across the penny-basic polymyxa, this is the tablet and penny-basic express, they can able to produce hydrophore also. Here is the picture of production of hydrophore. After that, I isolated that all isolates. I did the molecular assay of IPs, fungal isolates only. Up to now, I did only 6 isolates. Remaining isolates I have to do because of lockdown. Now started up to now the work. Now they are there. We have the little diversity. They are come from all the bacillomycetes, zygomycetes, all are coming from them. Then I straight away did field evaluation because we know to perform to check, evaluate them. So I took the 2 chemicals, I took them and the 5 fungal endophytes and the 4 bacterial endophytes and 1 bacterial Burkholderia arboris. It's already evaluated by IRI. It's performing very well in abiotics test for abiotics test. So I took also that also. And
Rajput, please conclude.
Okay, sir. 1 minute. In that we know that mostly the bacterial isolates they are performing very good rather than the fungal isolates. And here I can show the picture of the endophytes. The 2 fungal isolates, one of Aspergillus flavus and second one is Fusarium falciforme. They become pathogenic and they are not allowing to grow soybean also. It is the best performing isolates of Bacillus. And here my conclusion is that endophytic for soybean, endophytic bacteria they are performing very good for the management of foliar diseases rather than the fungus. And among the bacteria, Bacillus cereus and Bacillus acrensis, they are performing very good. Thank you for giving me for this presentation.
Thank you, Dr. Dhananjaya. Any one question, please? Quick question. Anybody? Any question? Clarification? Okay. If there is no questions, thank you, Dr. Rajput. May I request the next speaker, whoever is ready? Hello? Please mute. Others, please mute.
Sir, Dr. Dhananjaya, please.
Yeah.
ಊಟಲ್ಲಮ್ಮ ಇಲ್ಲಿ ಆನ್ ಇದೆ ಇಲ್ಲಿ ಇಲ್ಲ ಲಿಸ್ಟ್ ಆಗ್ತಾ ಇಲ್ಲ ನಮ್ಗೆ
Hello, sir.
ಯಾರು
Good afternoon.
ಮಾತಾಡ್ತಾರೆ?
Sir, the next speaker will be Dr. Ali Anwar, sir.
Okay. ಗುರುತಿಲ್ವಾ?
Sir, he'll be joining at 45, sir.
Okay. I will invite Dr. Ali Anwar. Ali Anwar, please.
Yes, sir.
Please go ahead with your presentation.
Okay. I am loading this slide.
Dr. Dhananjay, kindly mute. Dr. Dhananjay, kindly mute your phone.
Mute ಮಾಡಿ. Dhananjaya, please mute. Mute your microphone. Dr. Dhananjaya.
Yes, you are hearing. I am ready, sir.
Please mute. Dhananjaya.
Okay, sir. I ready to.
Go ahead.
Okay, sir. Audible and.
Dhananjaya ಅವರೇ ಮ್ಯೂಟ್ ಮಾಡಿ. ಮ್ಯೂಟ್ ಮಾಡಿ ನಿಮ್ಮ ಮೈಕ್ರೋಫೋನ್.
Okay, sir. I am audible.
Please go ahead, Ali Anwar.
Yes, sir. I am ready. You are audible.
Please go ahead.
Okay, sir. Thank you very much. Okay, this is my great pleasure, sincere pleasure, and to talk over the effect of trichoderma bioformulation against fungal foliar diseases of tomato under temperate condition of Kashmir. We have conducted the study over this aspect, and then we some interruption is going on. Okay.
Please, others.
Okay. In fact, tomato is an important. There is no need to tell this one. Well, is all over the world for its culinary properties. But production is affected negatively by number of biotic and abiotic stresses. Okay.
Kindly mute yourself, Dhananjay, sir. Yes, sir, you can proceed.
I am audible.
ಅರ್ಥ ಆಗ್ತಾ ಇಲ್ಲ, ಫೋನ್ ಮಾಡಿ ಹೇಳಿ. ಡಾಕ್ಟರ್ ಧನಂಜಯ, ಪ್ಲೀಸ್ ಮ್ಯೂಟ್.
Okay, sir, you can proceed.
Dr. Anwar.
Okay, thank you very much. The most serious biotic stresses are caused by diseases such as wilt, leaf spot, and blight diseases, which are prevalent in this valley. It is popular vegetable. Tomato basically very popular among the people of the Kashmir. Those are growing it, and they are suffering with these such diseases. Kashmir valley crop is grown over.
Hello, sir. Ali, sir.
Yes, sir.
Can you please put the slides on the slideshow mode?
Yes, I can. Okay. Basically, I was telling fungal rots are responsible for causing serious production problems and become winners of tomato in valley. Because of that, there are many diseases, Buckeye rot and late blight, early blight. Those are causing the fruit rot in the valley, and the farmers are unable to get a very good production and productivity because of these type of diseases which are occurring. And fungicide, those growers are using to control the diseases by fungicide, but they are not able to control completely, but they have needed substitute of the controlling measures. Due to that, we have taken the bioformulation of trichoderma to manage the disease. Actually, this is the satellite view of my trial, my university, where we have conducted the trial. And so there is a view of satellite. And in fact, material and methods which we have taken to accomplish this disease, this study. First, we have conducted this trial in our research field, where we conducted this trial, and basically we took the variety Shalimar 1, which is prevalent among the growers, and it is determinate nature of the variety. And basically, this Shalimar 1 was grown in nursery during the month of April for both the years, subsequent years. And then after 35 days of tomato seedling, which was become transplantable, and then we have carried it into the plot size 1.75 into 5 meter square with a plant spacing of 40 by 30 centimeter. 14 treatments of the bioformulation combination, those are here. Talc bioformulation containing the 10 or 6 CFU of the trichoderma, and another CFU 10:9, and simultaneously oil bioformulation was also taken containing 10⁶ and 10⁹ CFU, and both are based formulation. And we have taken the one chemical mancozeb 75 WP at the rate of 0.3% per liter of water. And the combination of those we have taken, and there are the combination.
ಗುರುದತ್ ಕೈಂಡ್ಲಿ
ನಿಮ್ದು ಮಾಡಿ ನಮ್ದು ಮಾಡ್ತೇವೆ, ಅವರಿಗೆ ಕೇಳ್ತೀನಿ ಯಾವ್ದು ಮಾಡ್ಲಿ ಅಂತ.
ಮ್ಯೂಟ್ ಕೈಂಡ್ಲಿ
ಗುರುದತ್ ಅವರೇ ಮ್ಯೂಟ್ ಮಾಡಿ. ಮ್ಯೂಟ್ ಯುವರ್ ಮೈಕ್ರೋಫೋನ್.
ಈಗ ನಿಮ್ದು ನೀವೆಲ್ಲ ಇದ್ರಿ ಸರ್? ಈಗ
ನೀವು ನೆಕ್ಸ್ಟ್ ನೀವು ಮ್ಯೂಟ್ ಮಾಡಿ ಫಸ್ಟ್.
ಹೌದಾ?
ಮೈಕ್ರೋಫೋನ್ ಮ್ಯೂಟ್ ಮಾಡಿ.
ಸರಿ ಸರಿ, ನಾ ಮಾಡ್ತೀನಿ ತಗೊಳ್ರಿ. ಓಕೆ,
ಮೈಕ್ರೋಫೋನ್ ಮ್ಯೂಟ್ ಮಾಡಿಯಪ್ಪ.
We have muted it. Okay, then we have taken the chemicals and made the combination with the bioformulation with the different concentration and with different CFU of the bio entities which is containing in the bioformulation, and we made 14 treatments which were carried under the randomized block design in our field. Talc and oil based bioformulation containing 10:6 and 10:9 CFU, which containing the trichoderma herzianum, and we took this bioformulation 5 gram in case of the talc based bioformulation, and 5 ml in case of the oil based bioformulation, and made it into the water and separately with the combination of the mancozeb 75 WP at the rate of 0.3%. 4 sprays. We have taken 4 sprays of the bioformulation at different stages of the crop in each treatment. All the recommended agronomical practices were done here, and we have left one unsprayed plots which was controlled, and this is my view of the university where we have conducted this trial. And these are the photographs of the different stages of the crop where we have conducted the trial. First is this shows that there is a view of vegetative stage of the crop where we have taken 14 treatments, and we conducted these trial. And we are making the bioformulation solution which we have sprayed over this stage of the crop, and then we applied all the agronomical practices and tried to beet-free trial, and then we have made some other cultural practices in the field to avoid all kind of the hampering to success the trial. And then we have made the staking of the crop, because of that when crop grow up and become fruit stage, and then we have to make some staking over that. Because of that, the vegetation or fruits should not come touch in the over the surface of the soil, So,
Dr. Ali, please
yes.
conclude. Please conclude.
Okay, okay. Thank you very much. We are coming over the result. When we have completed this trial, we found that septorial leaf is what caused by septorial lycoparsis, which is attacked and invaded the plants of this tomato. So the treatment, 13 treatment, which is mentioned here, and 25% we have to take in the chemical of mancozeb 75 WP at the rate of 3% per liter of water, included in this into the 75% oil-based bioformulation at the rate of 10:5 CFU. Then we control this Cercospora very excellent, and got this 52.87% in this with this treatment. And next, and exhibited a minimum disease severity 1.97% with the highest disease control, 74.97% early blight disease caused by Alternaria solanae by this treatment. And in case of late blight caused by Phytophthora infestans, a minimum disease severity 0.92% with the highest disease control, which is declined the incidence and severity of the disease by 68.81%. Whereas in case of Cercospora leaf is 5.35 with the highest disease control, 73, as it is shown in the table. All the combination of the treatment, which were found very efficacious, very efficient, and found significant control over the foliar diseases which are occurring in the field. And in general, all the treatments help in reducing the disease severity over the leaves when we compare with our unsprayed trial. I would get 25% chemical and 75% bioformulation, which was very 10 to power CFU. So it was very effective over the all kind of diseases. These are the photographs and the diseases which were found in our trial. And next, impact of the bioformulation, we have observed the data and recorded the incidence of the disease and severity, analyzed the severity of the diseases, which were found over the fruits. Data maximum showed, which are mentioned in this table, depicted in the table. 50% chemical, means we have taken the mancozeb, and 50% oil-based formulation at the rate of CFU 10:6. And 25 chemical, both treatment which were found very effective and declined the disease incidence and severity or by this these two sprays of the bioformulation. And the highest fruit rots were observed when the plot was not treated with any combination. Therefore, we consider that the combination of the treatment are most effective by reducing the fruit rot, resulted the highest fruitage. But similarly, we have in the same experiment pertinent to evaluation of BKC, different combination of bioformulation was found. Similar trend, the major diseases of this bioformulation. Economic, we have also analyzed the economic analysis we have done at this trial and found here the very good result of that. The following economic analysis has been based on the fact that no rental was paid to the university on account of land, make success conducting field experiment during 2020. So here there is a list of the inputs which was occurred during the conduction of the trial. Here acknowledgement, I thanks to Dr. Nidhi Didwania, who is managing director of Agronica Private Limited, Faridabad, Haryana, from where we have got this bioformulation to validate against the foliar diseases of the tomato. And I also pay the thanks to Director Research of our university, who has facilitated us to make the success of this trial very adequate manner, and what we have presented here. Thank you very much, sir.
Thank you, Dr. Ali.
Thank you.
Since you have taken 5 minutes more, no questions here. Anybody personally, you can clarify. Next speaker, Gurudat Thegde, ready? Dr. Gurudat Thegde, ready? Hello, Gurudat Hegde. If he is not there, Nandan. Nandan,
I am ready, sir. Yes, sir.
please
Gurudat
go
Hegde.
ahead. Start. Nandan, please start.
Sir, Hegde is there, sir. Dr. Hegde is there.
See, he is not responding. He is
No,
not
no, no. He's
responding.
there. Sir, he is muted. Unmute, Dr. Hegde, please. Unmute and start.
Sir, Gurudat
Gurudat is there?
Can I? I'm
Gurudat
here.
Hegde? Gurudat Hegde? Please continue. Please continue. Please continue.
Sir, audible, sir.
Yes, sir,
Continue, please.
Audible, sir. Sir, can I start?
You are audible, sir. You are audible, sir. Please continue.
Okay, sir. Okay, madam. Good afternoon, everybody. Respected chairman, sir, co-chairman, and all the distinguished national and international delegates on this international conference. I'm just going to give a brief account of my research on potentiality of the biopesticides against the fungal foliarities of vegetables grown
Hello,
organically under protected.
Sir,
Hello, sir.
Gurudat,
Hello, madam.
we are unable to see
Hey.
your slides. Okay. You're sharing still.
I already shared.
Now it is visible. Put it on to slideshow mode, sir.
Slideshow mode only, madam, now.
No, sir.
I have kept it in slideshow mode only.
Okay. It is not. It's not coming.
I have kept it in slideshow mode.
Okay, sir.
Otherwise, I have to stop sharing and again I have to reconnect, reshare. Shall
You
I?
can please do that, sir. Reshare it.
Chairman, sir, 1 minute, sir.
Sir, now it is visible, sir.
Now visible.
Now visible. Yeah. You can continue, sir.
Okay. Okay. Respected chairman, co-chairman, and all the distinguished delegates, I am going to give a brief account of my research on potentiality of biopesticides against fungal foliarities of vegetables grown organically under protected cultivation. See here, there are three different types of concepts are here. One is use of biopesticides, organics, and also under protected cultivation. And this is our bioresource farm where it is organically certified land, and the protected cultivation. I have taken disease management in tomato and cucumber. And we all know the importance of organic farming because there is a huge demand is there for organic products for the safe and healthy food. The ill effects of plantation, the plant protection chemicals on the flora, fauna, humans, and environment as a whole, they are the major concerns in inorganics or the overexploitation of inorganic chemicals. And therefore, we are switching this last 2 decades, we are switching slowly over to the organic farming. And protected structures, we know that the main purpose of the protected cultivation is to create favorable environment for the sustainable yield as to realize its maximum potential. So keeping these things in mind, we have went to the organic cultivation of these two major vegetables for a better quality produce, higher productivity, off-season cultivation, better insect disease management, efficient use of resources, and the ever-increasing population, decreasing the land holding, increasing pressure on natural resource, land and water, we went to this protected cultivation. Nowadays, we all know that protected cultivation, soilless culture, hydroponics, etc. are evolving as a new modern concept for the effective management of pest and diseases and to increase the productivity. We know that after having so much of importance of these biopesticides, only there are 4% consumption in the global market. So we have to create awareness among the farmers and the scientific fraternity to go with more of organics, to use of more and more biopesticides to manage these fungal diseases. And I'm not going very much detail into this introduction part because we all know tomato is one of the richest vegetable crop consumed all over the world. And this is the scenario of world, India, and also in our state. The best way to increase the production and also productivity of vegetable is growing the crops under protected cultivation. One of the expert in the protected cultivation, Brahma, he has given his statement. And we are after these PGPR and biocontrol agents all over now to minimize the loss of or to minimize the use of chemical pesticides. And these microbes, they are not only important as a biopesticides, but also as a biofertilizers. And coming to straight away to research, see, for any organic cultivation, the soil entity is very much important. So we have given a very importance to the soil fertility in the organic way by mixing of neem cake, vermicompost, rock phosphate, and FYM. And these are the different treatments, the treatment details, soil treatment with all this organic matter, and Panchagaya that also we have used through, and biofertilizers, trichoderma, pseudomonas, PSB, Pasteuromyces, these things are also used as a nematode management, also to manage some of the soil-borne diseases. And myco-insecticide were used Dimoria, Lecanicilium to take care of some of the pesticides, pests. And you can see the clear view of the organically grown tomato under protected cultivation at various growth stages. And we can see the traps, spore trap, as well as also sticky traps that are also being used to manage the insect pest. And we all know, as pathologists, we know these are the major diseases under protected cultivation as well as under the natural condition. And we are successfully managing these things under through biopesticides. We can see not only on the leaf, but also on the stem, various spots, stem folium, alternaria, and these are the pathogen structures. And these are the different treatments combinations that have been used under protected cultivation. Seed treatment with trichoderma and seedling dip with trichoderma, followed by spray with trichoderma. Again, Bacillus subtilis, seed treatment, seedling dip, Pseudomonas. Pseudomonas also in the same condition, which is compared with recommended check, that is sulfur, because we all know that sulfur is recommended, you can be used under cultivation, which has been compared with the untreated control. And you can see among the different treatments, treatment number 2 and 3, wherein we have Bacillus subtilis as well as Pseudomonas fluorescens as seed treatment, as a seedling dip, and 4 sprays at 15 days interval has considerably reduced all the fungal foliar diseases, and which has been reflected in the yields also. There are different mechanisms works out when we use these biological control agents, antibiotics, and mainly it is a induced cystic resistance. Mainly we say when you use it in the field conditions. So as far as the cost economics, say for anything organics or inorganics, ultimately farmer requires the cost economics. So we can see even the cost in the treatment number 2 and 3, it is 3.22 and 3.18 with the net returns of 6,06,250, 6,19,240 per hectare of this tomato under protected cultivation. So this is very much important as far as the farmers' benefit or farmers' concern is concerned. And not only this yield increase or disease reduction, but also the plant parameters. We can see plant height, number of fruits, number of clusters, plant height, all things have increased significantly compared to the untreated control. And you can see the number of pickings. So I would like to very stress here, number of pickings also more in case of this organically treated plants. So go green technology. Now people are talking about more and more yield in a limited space. Wherever you have used the Pseudomonas bacillus, it has triggered the plant growth hormones and has increased the pickings. We went up to 12 and 13 in case of this BGPR treated treatments as compared to the check and also untreated control only 9 and 10. And we can see the difference between the sulfur and the Pseudomonas fluorescens. You can see the totally unspread plot versus Pseudomonas fluorescens. And this is a typical organically grown tomato fruits, which is a indeterminate type of variety. And our Honorable Vice Chancellor, many of the dignitaries, they have visited this protected cultivation, and then they have given suggestions to improve upon the cultivation. Dr. Subramanya, and you can see Honorable Vice Chancellor M.K. Naik, S.A. Patil, our Vice Chancellors, they also visited. Many of the foreign delegates, our own alumni, they have also visited during the course of its investigation. Dr. Shrikant Kulkarni, V.B. Nargun, probably most of you know these experts who have given a lot of inspiration for me to conduct these experiments. You can see Dr. Mista, Dr. M.B. Patil, and Brahma Singh. I just in the beginning I mentioned Brahma Singh.
Sir. Gurudat sir, we are able to see the PPT of the table, effect of treatments with promotion. Maybe the slides are not moving.
Or here it is moving, madam.
Our visibility is there is one table saying the effect of treatments with different bioagents or something. Can you please look
Continue, madam.
this?
Let him continue his presentation.
Okay, sir.
Let him continue.
Okay, sir.
Okay.
Sir and madam, I am just continuing with the slides. It is moving here. So, Professor Brahma Singh, he was also very keen in observing some of the diseases in the protected cultivation. And not only these biopesticides, we have even used this garlic, cow urine, and vegetable sulfur in combination to manage some of the fungal diseases. This is a part of my students' work, which she has carried out during her MSc program. And we can see treatment number 7, the decrease in the disease incidence of early blight, septoral leaf spot, and gray leaf spot of the tomato. And we can see the difference between the integrated approach, Pseudomonas, cow urine, and vegetable sulfur against the untreated control. And this is the difference between the untreated as well as the PGPR treated plots. And you can see the plant parameters, internodal length and plant height also increased in PGPRs. And as far as the cucumber is concerned, the downy mildew and powdery mildew are the major problems. Downy mildew and powdery mildew, these are the major problems. You can see the severity of cucumber powdery mildew as well as the downy mildew in the protected cultivation. But here you can see the treatment number T5, the consortia of two PGPRs, Pseudomonas fluorescens and Bacillus subtilis, has significantly decreased the powdery mildew and downy mildew, and which in turn has reflected in the increase of the cucumber plants. And just we can see comparative to the untreated control also. And as far as the cost economics is concerned, Pseudomonas fluorescens combined with mixed with Bacillus subtilis, that is 5 grams 5 gram, has also increased the net returns and the cost benefit ratio of 3:80 as compared to untreated control. You can see this is a demonstration block wherein we have taken up the large scale demonstration by using these microbes to manage powdery mildew and downy mildew of the cucumber. This is a large scale view of the combined spray of the field cucumber. You can see some of the scientists, Professor Jagirdar, Ms. Patil, and Ms. Srinivas, they have witnessed this. And we can see the fully grown plant with enough number of fruits on the plant. And this is the difference between the Pseudomonas fluorescens plus 5 Bacillus subtilis sprayed as well as the unsprayed, untreated chuck under.
Dr. Hegde, please conclude. Dr. Hegde, please conclude.
Cultivation of organic vegetable under protected cultivation, and which acts as a plant growth promoting rhizobacteria, and stay green technology can be very well managed through by the use of biopesticides under organic cultivation. And there is a need to develop a basket of technology options for sustainable agriculture practices that amalgate traditional knowledge with modern science-based practices. We have to ensure the availability of quality bioproducts to help the farmers to practice organic farming practices. Finally, I acknowledge US Dharwad and University of Medical Sciences Bagalkot for giving me an opportunity to present as part of my research work at Dharwad. Thank you very much, sir.
Thank you, Dr. Hegde. Next speaker, Dr. Nandan. Nandan is there?
Yes, sir. There, sir.
Please go ahead. Please go ahead. Please start.
Gurudutt sir, please end the presentation, sir.
Enama?
End the presentation, sir. You should end the presentation.
Yeah, let him do that.
Ended, sir.
Be
I have closed.
Hegde ಅವ್ರದ್ದು close ಮಾಡಿ.
Sir. I have closed the.
It is taking little time. It's okay.
Is it okay, sir?
Sir, or you exit, sir?
Kindly exit.
Kindly
Yes,
exit.
yes. I think it is okay now, ma'am. I have come out, sir.
Sir, Nandan should start presenting, sir.
Sir, I have already told him to start.
Yeah, madam. 1 minute, madam. It's not sharing right now. Madam, is it visible, madam?
It's showing none. It is visible. Put it onto slideshow mode.
Is it okay, madam?
Yeah, please go ahead. Please go ahead.
Okay, sir. Respected teachers, seniors, and my dear friends, a very good afternoon to everyone. Myself, Nandan, PhD student, Plant Pathology, US GKVK Bangalore. Today, I am presenting part of my PhD research work in this conference. Before going to actual topic, I would like to give you a brief introduction regarding onset of this research project. The slides are not moving right now here, sir. Yes, sir. Okay, sir. Fine. So before going to actual topic, I would like to give you a brief introduction regarding onset of this research project. In agriculture, at present scenario in agriculture, plant diseases will going to have a huge threat with respect to the crop product and also productivity. And if you consider a tomato crop system, the foliar diseases like a powdery mildew, early blight, many of the diseases will going to have an impact on yield loss. And if you're going to consider some of the soil-borne diseases like a rhizoctonia, root rot, sclerosis wilt also having the same impact. So generally, to manage this kind of the diseases, soil-borne diseases, we're going to follow the cultural practices, chemical drenching, biocontrol agents, etc. So at this point, what we thought is, why can't we encounter this particular soil-borne diseases in tomato crop system by using the hidden world living organisms? So what are all these hidden world living organisms is the question for us. So if you just consider our human body itself, we will going to have a n number of microorganisms inside our body, where it will going to have a beneficial processes like it for helping in case of digestion, immunity, many of the things. As a similar kind, the plants also will going to contain a living organisms that will going to impart a stress tolerance against the plant pathogens. Those are all nothing but fungal endophytes. So it may be a bacterial endophyte or maybe a fungal endophyte, but we are concentrating on the fungal endophytes. Means to explore these fungal endophytes against the rhizoctonia and sclerotium pathogens in the tomato crop system. So by considering all these backdrop, we put forth a thesis title, our title as antagonistic effect of the fungal endophytes against sclerotium and rhizoctonia species infecting the tomato with the following objectives, that is the isolation, screening, and disappearing the mechanism of action. So as a first part of our work, we have isolated the fungal endophytes from different weed species, that is the Tridax, Cassia, Parthenium, Pongamia species, and by following the standard fungal endophyte isolation protocol method on the potato dextrose agar media, that is from the leaf tissue, stem tissue, and also root tissues of the different plants. So from these 4 different plants, that is Tridax, Cassia, Parthenium, and Pongamia, we have obtained total 43 fungal endophytic OTUs, that is 14 from the Tridax species, 10 from the Cassia species, and 3 from the Parthenium species, and 8 from the Pongamia species. So total we have 43 fungal endophytic OTUs. So next thing is with respect to the pathogen. We have collected the tomato samples infected with the sclerotium pathogen and rhizoctonia pathogen in the farmer's field and processed it for the pathogen isolation on the potato dextrose agar media by surface sterilization them with the 1% sodium hypochlorite solution. So the pure culture of the pathogen has been obtained and it has been morphologically confirmed through microscopic studies as the sclerotium species and rhizoctonia species. Later, these pathogens has been mass multiplied on the sorghum seeds and inoculated to the plants containing, that is the parts containing the tomato seedlings, to confirm the pathogen is virulence or not. So finally, after confirmation of the virulence, we took forward this pathogen for our further studies. So now we have 43 fungal endophytes as a fighters against these 2 enemies, that is the sclerotium and rhizoctonia pathogens. So how these fungal endophytes will going to get fight against these 2 pathogens? For this condition, we have used a platform called as a dual culture, where the 4 biological replicates has been used for the dual culture and 1 as a pathogen control. And the inhibition percentage of the pathogen has been calculated after 7 days of the inoculation. So here we can see the different inhibition percentage of the sclerotium rhamnosus and also the rhizoctonia pathogens by different endophytic fungal OTUs from a different plant species. So among these, there will be a differential inhibition percentage of those sclerotium and rhizoctonia species, but within these OTUs, there are 4 fungal endophytic OTUs that shows more than 50% inhibition of the rhizoctonia, and 2 endophytic OTUs has been shown more than 50% inhibition of the sclerotium species. So now the thing is, to which taxonomy these fungal endophytes and pathogen has belongs to. So just to check the taxonomy of the same, we have gone for the molecular characterization by genomic DNA isolation and amplification of the internal transcribed spacer region. So it has been amplified at 650 base pairs, where the sequence data has been flashed through the NCBI nucleologist nematode blast, and results revealed that the pathogen as Rhizoctonia solani and SRT as the Athalia rausi, that is the perfect stage of Sclerotium rausi, and the potential endophytic what you CSR1 as Macrophomina pseudophagiolina, CSR3 as Ceratobasidium species, PHR3 as Trichoderma aspergillum, and PPS3 as Aspergillus niger. So among these fungal endophytes, the Trichoderma aspergillum, which has been isolated from the Parthenium roots, is known to have a better inhibition action against the Rhizoctonia solani and also the Athalia rausi. So we took forward for this fungal endophyte for our further studies. So now the question is how this fungal endophyte is inhibiting the Rhizoctonia solani or the Sclerotium rausi. Means to know the mechanism of action of this fungal endophyte against the respective pathogen. So first one is competition. Of course, just by looking into these plates, we can say that there is a more than 50% of the space occupancy has been formed by the endophytes against these pathogens. So we can say that there is a competition for space in vitro condition. So next, if you coming to the antibiotics, whether this Trichoderma aspergillum has an ability to produce the antifungal diffusible compounds against the Rhizoctonia solani or Sclerotium rausi, first we have extracted the secondary metabolites produced by the Trichoderma aspergillum by following the ethyl acetate extraction method. And this extract has been concentrated by using the vacuum concentrator, and to this crude extract we have added ethyl acetate as a solvent, and this solvent has been impregnated into the sterile disc. Means we have gone for a disc, that is the sterile disc assay in the potato dextrose agar media containing the pathogen. So that disc sterile disc assay results revealed that if you're just looking into these slides, we can say the carbendazim 0.1% as a positive check, the ethyl acetate as a negative check. Just by inoculating the 20 microliter of this solution, there is no any inhibitory action by the carbendazim. But if you're just looking into these slides over here, there is a crude extract of the Trichoderma aspergillum, which shows there is a aggregation of the mycelium exactly at the where we placed the disc. So it shows that there is some opposition made by this crude extract against the Sclerotium rausi. And if you're just looking into another picture over here, where there is a clear-cut inhibition zone has been formed by the crude extract of the Trichoderma aspergillum against the Rhizoctonia solani. Here we can see as a positive check 0.1% of the carbendazim solution. So here we can see the negative check as a ethyl acetate, there is no any inhibitory activity. So just by looking into these results, we can say that the Trichoderma aspergillum has an ability to produce the antifungal diffusible compounds against the Sclerotium rausi and also the Rhizoctonia solani. So next, coming to antibiotics, see volatile compounds, whether the Trichoderma aspergillum has an ability to produce the antimicrobial volatile compounds or not is a question for us. So just to check the same, we have gone for the double petri dish method, where the pathogen has been inoculated on the upper lid of the petri plate and endophyte on the lower lid of the petri plate, and we make them into an airtight condition, and there is no any physical contact between the pathogen and endophyte has been made sure. So this results showed that there is a 52.2% inhibition of the radial growth of the Sclerotium rausi and 26.66% inhibition of the Rhizoctonia solani in the double petri dish assay. So just by looking into these results, we can say that the Trichoderma aspergillum has an ability to produce the antimicrobial volatile organic compounds against the Sclerotium rausi and Rhizoctonia solani respectively. So further, if you just earn the name that is the Trichoderma, its ancestral property that it will going to have some hyperparasitic behavior against many of its antagonistic fungi. So we have gone for scanning electron microscopic analysis of the interaction between the respective pathogen and Trichoderma aspergillum. So the results revealed that if you just look over here, there is a coiling of the Trichoderma mycelium and also colonization of the single spores, single cell spore, that is the Trichoderma spores, and the mycelium of the Rhizoctonia, where it will going to secrete some of the lytic enzymes and finally lead to lysis of the Rhizoctonia mycelium. And in the second picture, we can see that the sclerotial body of the Sclerotium rausi has been parasitized by the Trichoderma mycelium, where the sclerotial body will going to get decayed and died. So just by looking into these pictures, we can say that the fungal endophyte Trichoderma aspergillum has an ability of hyperparasitic behavior against the Rhizoctonia solani and also the Sclerotium rausi. So next thing is colonization assay. So why this colonization assay required here means because we have isolated the fungal endophyte some of the weed species, and we are interested to get colonized in the tomato plants. So to follow the colonization assay, we have treated the pre-germinated tomato seeds with spore suspension of the Trichoderma aspergillum for 3 hours, and the plants have been grown up to 25 days. So after 25 days, these plants have been harvested and processed for fungal endophyte isolation by following the standard protocol method, and the results revealed that there is a successful colonization of the endophytic Trichoderma aspergillum in the stem and the root tissues of the tomato plants, but it is not at all colonized in the leaf tissues of the tomato plants. Since we are dealing with the soil-borne diseases where the pathogen will going to infect at the collar region or root region, but the Trichoderma aspergillum is present in both stem and also root, so there is a better platform for fight against these pathogens. So finally, I would like to conclude my presentation that we have identified some of the potential fungal endophytes against Sclerotium rhamnosus and Rhizoctonia solani, and also they are showing better competition in the in vitro condition. And one of the fungal endophyte, that is the Trichoderma aspergillum, is known to produce antimicrobial diffusible and also antimicrobial volatile organic compounds against the Rhizoctonia and Sclerotium species, and also it possesses hyperparasitic behavior against the Rhizoctonia and Sclerotium pathogens, and it is also successfully colonized in the roots and also stems tissues of the tomato plants. So now the big question mark right now here is whether the same endophyte will going to have a same efficacy in the field condition or not is a big question for us. So right now we are following the same in vivo studies right now. So that is the progress. The work is in progress. The remaining work is in progress right now. So thank you everyone for giving me this opportunity to present my PhD work in this conference.
Thank you, Mr. Nandan. One question, please. Any one question only.
Yes, sir. Yes, sir. Please, sir.
One good question. Okay. If there are no questions, may I request Divya Mishra? Miss Divya Mishra is there? Divya Mishra? If not there, Miss Chaitra ready? Chaitra?
Yes, sir.
Please go ahead.
Hello.
Please.
Okay, sir.
Nandan. Okay. Chaitra, start sharing the screen.
Yes, sir. I'm sharing.
Password 1 2 3 5.
Sir, is it visible, sir?
Yeah, go ahead. Go ahead.
Good afternoon, one and all. Respected chairman and co-chair. Now I am presenting my research paper on profiling secondary metabolites of endophytic fungi isolated from cocoa and its anti-humectous activity.
Chaitra, kindly put it on.
Sir?
Kindly full. Yeah, that's it.
Chaitra, put it on to slideshow mode.
Slideshow, please.
Now it is visible, sir. Hello? Hello?
It is audible, it is visible. Go ahead.
Okay, sir. Coming to the introduction, cocoa, it's called as food of God. It is an beverage and industrial and as well as plantation crop, mainly grown as intercrop in arecanut, coconut, and oil palm garden. And it's also grown in partially cleared forest areas. Economically important diseases in cocoa are black pod rot and stem canker diseases. These two diseases are caused by the pathogen Phytophthora species. This disease, it can cause an yield loss ranging from 10 to 100% depending upon the weather condition. When it comes to secondary metabolite, these are all organic compounds produced by fungi, bacteria, or plants, which are not directly involved in the normal growth and development, but they are very much required for survivability of this organism. In my in my research, I isolated 39 endophytic fungi from geographical area from South India, and they were screened against Phytophthora parvivora, ALB3, a virulent isolate causing black pod rot disease in cocoa. From out of 39 isolates, 4 isolates, that is a Diapothis species VF3 and a Coltotrichum species TN92, Arthrinium resicra windri P11 and Arthrinium resicra windri P12, they exhibited maximum mycelial inhibition in deer culture assays. So, crude metabolites from these endophytic fungi were extracted using ethyl acetate method, and they were screened against Phytophthora parvivora under in vitro condition. This is a photograph. Here we can see the different symptoms produced by Phytophthora in the cocoa plant. This is black pod rot and leaf blight, and this is stem canker. A photograph of endophytic fungi. This is Phytophthora parvivora, that is the pathogen which is used in this investigation, and Diapothis species and Coltotrichum species, and Arthrinium P11 isolate and Arthrinium resicra windri P12. This is the photograph showing their plate assay. Here, in one corner of the plate, the pathogen was inoculated, and the other opposite side, the endophytic fungi was inoculated, and percent inhibition was measured. And here we can see the inhibition by of pathogen by these endophytic fungi. Next, this is a flow chart of ethyl acetate method of secondary metabolite extraction, where where endophytic fungi was grown in malt extract broth for 7 days. Then the extract was collected, centrifuged at 6,000 RPM for 20 minutes, and the extract was acidified to pH2, and then it was extracted using ethyl acetate by using funnel extraction method, and it was concentrated by evaporating that ethyl acetate under room condition and dissolved in HPLC grade methanol. For agar-well diffusion method, these concentrated metabolites were dissolved in 1 ml of ethyl acetate. For GCMS analysis, it was dissolved in 1 ml of HPLC methanol, HPLC grade methanol. Then coming to agar-well diffusion method, here different concentration of crude metabolite were used, 10 microliter, 20 microliter, 30 and 40 microliter. Here and incubated for about 48 hours under room temperature. And here you can see the mycelial lysis activity by these crude metabolites of these endophytic fungi, indicating it is effective against this Phytophthora parvivora. Next, coming to metabolic profiling of endophytic fungi, that is Diapothis species VF3 isolate. In each isolate, 40 different compounds were detected in GCMS analysis, and among 40 compounds, the major compounds were N-hexadecanoic acid, and 9-octadecanoic acid and octadecanoic acid, and this is the GCMS result, graphical representation of that GCMS result. That peak represent percentage area coverage by this particular compound. Next, coming to metabolic profiling of endophytic fungi, Coltotrichum species isolate TN92. The major compound in this endophytic fungi detected were L-glucose and octadecanoic acid. And this is the graphical representation of this GCMS result. Next, coming to metabolic profiling of endophytic fungi, Arthrinium resicra windri P11 isolate. The major compound detected in this endophytic fungi or N-butyl L-homoserine lactone, and 3-furanoacetic acid, 4-hexyl, 2,5-dihydro, 2,5-dioxone, and ethyl AD glucopyranoxide and atrophinyl. And this is the graphical representation of that DCMS data. Next, metabolic profiling of endophytic fungi, Arthenium resicula windrei, P11 isolate. The major compound detected were 3-furanoacetic acid, 4-hexyl, 2,5-dihydro, 2,5-dioxone, and ethyl AD glucopyranoxide. This is GCMS graph. Next, these compounds were further statistically analyzed using using JMP software. The statistic tool used was principal component analysis. In this few compounds, 12 compounds were detected, were identified as principal compounds present in secondary metabolite, and the plus indicates presence of these compounds in particular endophytic fungal metabolite, and minus sign represent absence of these compounds in these secondary metabolites. And this here you can see the property of these chemical organic compounds and references. Among 12, few compounds like octodecanoic acid and hexadecanoic acid and palmitoleic acid, 9-octodecanoic acid and furanoacetic acid isomaltol, they showed they reportedly having antimicrobial activity against. Then coming to this is the graph showing different compounds. The principal component 1, these are all the principal component 1, and this accounted up to 35.8% of total variability, and principal component 2, they accounted up 3.9% of total variability. Next, coming to the this is the graphical representation of these compounds. Dark and this is the pictorial representation of this compound, where present in different endophytic fungi. This is the chemical structure of identified principal components. Next, coming to conclusion, from my study, it shows that some compounds like octodecanoic acid and hexadecanoic acid, palmitic acid, 9-octodecanoic acid, and benzoic acid, 4-hydroxy methoxy methyl ester, and 3-furanoacetic acid. These compounds, maybe presence of these compounds in that secondary metabolite might have contributed for mycelial lysis in Phytophthora. So these compounds can be used for further characterization. Thank you. Hello.
Thank you, Chaitra. Any questions, please? One or two questions. Any quick questions? Okay. No questions. Thank you. Now I request my co-chairman to offer
Hello,
his remarks.
sir. Sir, Krishnappa, sir. Krishnappa, sir. Sir, Dr. Shamrao Jangedar is sir is not there. So instead of him, Dr. Gurudat will be presenting, sir, since he is the co-author of the paper.
Yes, no problem, madam. Let him come. Let him start.
Gurudat Tegde, sir.
Yeah, you can start.
Yes,
please start.
You can share your slides and start presentations.
Okay, madam. Sir, is it visible?
Please go ahead.
Good afternoon once again, respected chairman, co-chairman, and all the distinguished delegates. On behalf of Dr. Shamrao Jangedar, I am presenting this invited talk since I am associated with some of his work. So his topic is on trichoderma, a potential millennium microbe for sustainable management of field and horticulture. Hello.
Hello, sir. The slides are not visible. Now it is visible, sir. Yes, sir. Put it onto slideshow mode and present. Put it onto slideshow mode and present.
Yeah, proceed, madam. Proceed.
Okay, you can proceed, sir. You can proceed.
So this is the topic, the trichoderma potential millennium microbe for sustainable management of field and horticulture crop of diseases. And we know this is a 2.5, 3 decades old, one of the model biofungicides. Still they are being used as compared to the other biofungicides. This is still a model biofungicide. And we all know that human-induced global changes. There are so many reasons why these global changes are occurring. These are the different reasons. Land use changes, habitat destruction, burning of fossil fuels, air and water pollution, temperature rise, elevated carbon dioxide, more precipitation, drought, sea level rise, more fires, ecosystem breakdown. Many things are contributing to the global changes. And now we have to we have to move forward with different kinds of organics or biopesticides to make the crop more productive and soil more fertile. And we know this not only our India, but also in our other countries, this doubling of farmer income is also another important step in every country. In this regard, there are three important systems, that is calorie deprivation, protein deficiency, micronutrient deficiency. We have to overcome these some of the obstacles to make our crop more productive and soil more fertile. Yesterday, there was a session on climate change and plant disease. So, strong influence of weather on plant disease, in particular temperature and humidity. And also the new crops and cropping systems are coming up now in the change. So new diseases are occurring. So to combat all these things, we have to ready with different kinds of new molecules, maybe organic molecule or new organic molecule, specifically for soil-borne diseases. This trichoderma with different types of species are there. We have to be consider these as one of the component in the disease management. Now, to our understanding about trichoderma, as most of the speakers just now, Nandan and MS Rao, Nakiran, many of the stalwarts have spoke about, given their lecture on mechanisms. This micro associated activated the antioxidant enzyme activities, and so many PR proteins have been released, and through which the plants get triggered, and most of the disease have been under control. And we all know this. There are so many mechanisms that work out not only because of the trichoderma, but also other organisms like PGPR. There is a competition, antibiosis, induced resistance, hyperparasitism. This takes place, and we all know there is a competition and coiling mechanism that takes place whenever you use this trichoderma, both in vitro and as well as in vivo conditions. Now, coming to the trichoderma research trends, you know, this transcriptomics and development of transgenic plants are present day talk of every research work. Application of molecular tools in genotypic characterization, tagging of GPFs, that is green fluorescent protein groups in ecological studies. And we all know that protoplast technology, which helps in strain improvement, consortia application nowadays, just like a combi molecules, like a combination of chemical fungicides, there are trichoderma based combi products are also available in the market. That is trichoderma pseudomonas, trichoderma bacillus, trichoderma pacillomyces, so that from a single product we can manage many of the diseases. As far as this transcriptome is considered, see, there are so many works has been done with respect to the transcriptome studies of this Trichoderma hergianum, not only in India and abroad. The present study provides valuable information for future studies on biomass degradation and contributes to better understanding of the role of the genes that are involved in the process. We all know that consortia or compost culturing, that is also a major point in the present day era. So therefore, trichoderma can be used as one of the decomposing organisms, hasten the process of decomposition. And another study, identification of mycoparasitism related genes against phytopathogen Sclerotium sclerotiana through transcriptome and expression of profile analysis in Trichoderma hergianum. Here we have the author revealed that the first initiative to use of this RNA sequence for identification of identity for Trichoderma hergianum strain TR274 in response to the phytopathogenic fungus Sclerotium sclerotiarum. We all know that the expression of trichoderma genes in plant has benefited results. Many of the pathogen including the soil-borne diseases, have been controlled. We know that co-expression of endo, that is ECH42 and exo, NAG, chitin, they are responsible for producing so many PR proteins, as well as this multiple expression of the rice transgenic encoding 2 chitinase genes are responsible to manage or to induce resistance for Rhizopteris solani and Magnifor tigricia. And similarly, expression of Trichoderma hergianum, including having 70 genes in Arabidopsis species, also has reduced the diseases. Like that, Trichoderma hergianum, THKL gene encoding a clutch repeat protein was also has been successfully tried in Arabidopsis species. Trichoderma, we all know this is a species widely used in agriculture, and still it is being referred as a model biofungicide. It has been looked after or been mass multiplied in various institutes and various industries as a model biopesticides. These fungi reproduce asexually by production of conidia and chlamydospores, and they are very efficient mycoparasites and prolific producers of secondary metabolites, and that is why it has been considered as one of the best mycofungicide to control many of the diseases. So many strains produce elicitors and induce resistance, and the comparison of primarily saprophytic species with the mycoparasitic species has provided striking constraints and established that mycoparasitism is an ancestral trait of this genus. And antibiotic mediated suppression is well known by many of the institutes, and it is really making a great importance in the field of plant pathology. And to be effective, antibiotics, it must be produced sufficient quantities near the pathogen to result as to consider it as a biocontrol agent. This is some of the studies that growth inhibition of Rhizopteris solani by the Trichoderma. By Pythium ultimum has been suppressed by the Trichoderma virens by producing different antibiotics under in vitro conditions. And for any mycoparasite to be very successful, we all know that it should grow rapidly and it should sporulate profusely. It should infect parasitics in all ecological range early before host is damaged, and it should have the capacity to weaken or kill the pathogen and capability to persist well, non-pathogenic to cultivated host plants. So this is all about Trichoderma and some mass multiplication. See, all the institutes are having their own protocols for mass multiplication of the Trichoderma. In US, Dharwad is also having Organic Farming Institute, wherein we are multiplying 13 different organisms. And for Trichoderma, especially molasses yeast extract agar, is one of the best raw material or one of the best substrate wherein we can multiply quickly in a larger scale. So for any soil-borne diseases or any fungal diseases, we have to go initially with this dual plate culture technique, and Trichoderma hergianum is successful in giving a very good result. And we have got many of the students are also working on this as a part of their studies to become a successful entrepreneur. And we are following these fermenter-based technologies. Now coming to some of the field-based applications are considered. Professor Jagirdar is working in a soybean crop, it's a decade, and DSB 20 this disease, and but anthracnose is a major problem. And wherever they have went with the Trichoderma-based seed treatment, they have found the significant difference with respect to the disease reduction, that is anthracnose. And we can see the farmers' participatory programs were arranged to showcase the importance of this IDM approach, wherein the seed treatment plays Trichoderma as playing a major role. See, these are different, they have made different modules like a biointensive module, chemical module, adaptive module, and also the control. We can see the adaptive module has given a very effective and found very promising. So you can see the seed treatment to Trichoderma, then followed by spray with pneumonia arili, then hexaconazole, Pseudomonas fluorescens. We can see the reduction in pod blight, rust, and purple shade stream in both Dharwad Center and Ugarkur Center, because this soybean trial was conducted in both Dharwad and Ugarkur. In both the regions, successfully we have obtained the result to prove that Trichoderma is one of the best biofungicide if it goes as a seed treatment, and also as one of the component in the integrated disease management. Not only this Trichoderma or fungal diseases, but many of the insect pests are also being under control. And as far as the economic analysis of both the stations, Dharwad and Ugar is concerned, we can see the BC ratio is better in M3, that is adaptive model, wherein this Trichoderma Pseudomonas has been integrated with other modules and it has been successful in getting the highest costs. We can see the biointensive model, chemical module, control as if this adaptive model. It is very clear from this picture that adaptive model, wherever these bioagents have been used, the shiny growth and the very luxuriant growth canopy of the soybean crop. This is a demonstration from the farmer's field. These are some of the seed production plots wherein anthracnose disease, which is seed-borne, has been controlled successfully with the trichoderma. And we can see the dramatic changes in farmer that he brought the new tractor for his field operations because of the higher increase in the yield due to the lesser disease incidence. And these are some of the photographs wherein the soybean entire team went to different stations and demonstrated. Even the director from IISR Indore also visited such a demonstration block wherein they can have seen the significant difference of this adaptive model for successful management of the most of the diseases, including the seed-borne disease. So these are some of the extension programs so that they can create the awareness to the farmers. And these are the symptoms. And we can see the treatment T1 seed treatment with carboxyl and see the T4 seed treatment trichoderma harzianum, and which wherever T4 that is seed treatment is there, followed by further sprays with the chemicals, there also we can see the lesser PDI and more seed yield in all the blocks. See, there is a significant difference between the two treatments. One is control, another is seed treatment with trichoderma 6 gram per kg, followed by trifloxin and tebuconazole spray. Again, the similar, and not only in soybean, but in crops like tomato, banana, black pepper, sorghum, tobacco, soybean also, successfully this trichoderma and other ITKs has been used and has been given good result. And during 2000 only, this Panama wilt has been published in Plant Disease Research, wherein the trichoderma is one of the potential biocontrol agent. We can see soybean rust management even through trichoderma, cow urine spray and trichoderma spray at 0.5%. We can see the difference in PDI with respect to this treatment as well as the control. It is nearly 87-88% and as against 35% in trichoderma treated plots. And even the economics also has been very promising in this trichoderma treated plots. And as far as the PR proteins are concerned, you can see the upgradation or the higher level of peroxidase in the both peroxidase and polyphenol oxidase in the treated plots. So these are the integrated plots. Coming to the chickpea wilt, again one of the major soil-borne disease especially during the Rabi season, we can see these are the infected plots or the severity of chickpea wilt. And wherever it is treated with the trichoderma very systematically, soil application as well as the seed treatment, we can see the difference between the two plots, the wilt incidence in both treated and untreated plots. And this is one of the studies Professor Jagridhar conducted when he was there in Korea for his postdoc. When he was working with on the soft rot of tobacco, we can see the treatment 1 to the fourth one or fifth one, B4 plus 0.1 milliliter BTH, wherein they have used the biocontrol strain as well as the elicitor. There we have seen the complete reduction of this soft rot. That means as compared to the biocontrol strain alone and elicitor alone, and which is reflected in gus expression. We can see the highest expression of this gus, wherein this elicitor come biocontrol used in combination compared to the sole application. You can see that there are different kinds of products are available in the market with the different trade names. And apart from these studies, there are other studies that have also been reviewed in presentation elsewhere, not only in India but other countries on banana, black pepper, tomato, tobacco, pineapple, cucumber. Some of the technology transfers are concerned and our university work is concerned. Even sheath blight management is also being effectively done by using seed treatment with trichoderma harzianum. We can see the seedling dip with Pseudomonas, trichoderma, as well as azospirillum, which will enhance the seedling growth as well as reduction in sheath blight. Even in maize crops, seed treatment is recommended. Some of the oilseed crops, like you can see, the leaf spot and this sclerotium rot, trichoderma is very well in use by the farmers and it is giving a very promising result. Even in sugarcane, set rot management is well done by the trichoderma harzianum. Tobacco damping off, wherever we are using trichoderma as a sole or as an integrated, it will give the very effective result in tobacco. Similarly, in chili fruit crops and also in vegetables also, wherever used. In my presentation, I have shown you against tomato and cucumber. There also, wherever you have used trichoderma as a sole or as a consortia with Pseudomonas fluorescens, we have effectively managed some of the fungal foliar diseases. Even disease of mango, anthracnose, pomegranate blight. Morning, some presenters were talking about pomegranate blight. Can also be effectively managed even both as a soil application with trichoderma as well as the spray formulation which trichoderma manage the. So we can see the very healthy and bigger sizes of pomegranate wherever we use these biopesticides. And so even this is a well-known fact that our department has a history about this Sclerotium roxae in Dharwad and parts of Hasana in Karnataka. Also, this potato is being grown, and this is affected by this collar rot. Sclerotium is the fungus, and wherever we have used this potato treatment or feed tuber treatment with trichoderma, effectively managed. Yes, even we are educating the farmers how to multiply at in the organic matter, in neem cake or in FIM, how they can be used at the farmers level and they can be shifted to the farmers. Even in some of the coastal and Malnad areas, this areca husk are being used to mass multiply this trichoderma. We know this is one of the major problem in Western Ghats or Malnad areas, betel nut, areca nut, which is suffering by Phytophthora rot, or it is called as Scleroga in Kannada. So this is also being very effectively managed since olden days by soil application with this trichoderma in the followed by mulching. We know this black pepper, again, a slow wilt, quick wilt disease caused by Phytophthora. This can also be managed by the soil application of FIM, neem cake, trichoderma, that is native trichoderma is very effective in most of the cases. Then plastic mulching. We can see a farmer is applying neem enriched trichoderma all along the rhizosphere of the black pepper to manage successfully the wilt of this black pepper. And we can see the fast decomposition of the areca husk or the decomposing compounds because of the trichoderma. As in the beginning I told you, it is not only a biopesticide, but it also degrades some of the raw materials very fast, and we can see the bushy green appearance and the tuft of white roots coming out of this black pepper. And you can see the difference between the local method as well as wherever the mulching we have practiced trichoderma has successfully given the promising result. And in the ginger is also again soft rot or rhizome rot is a major problem. There also we have this trichoderma has been used successfully. In our university, the facilities available even in the open top chamber, because of rise in carbon dioxide, we are seeing the efficiency or effectiveness of this trichoderma on the open top chamber. The research is going on, and these are some of the protected cultivation photographs. We can see the consortia of Trichoderma pseudomonas in the middle layer, where the flowering is very profuse, very luxuriant growth. And of course, I told you this is like a green go-green technology. As you feed, it will grow. So you can see up to 4 or 5 months crop. We can harvest nearly 7 to 8 pickings in cucumber and 13 to 14 pickings in the tomato. And we all know that these are some of the success for biological control. Selection of the virulent strain is very much important. As far as possible, the native isolate is very much important. To go always as an integrated biological control agent rather than applying it as a sole. Then these are the futures of Trichoderma species, and the response are isolate specific and host specific. Publishing of isolate specific applied results will be more beneficial than the sole. Then potential for genetic trichoderma is people are working on it, and this should go as integrated management component, and low tech system as well as high tech. Immense value in extension services are there, and see bridging the know how to go or how to fill up the gap. Always as a scientist, we should think all this lab to lab, that is participatory approach, lab to land translational research, land to lab farmers wisdom, and land to land farmers to farmers listening. This is very much important, and we know that the FAO concept of FFS, Farmers Field School, has been very much implemented in most of the crops, wherein farmers have given equal chance to see the efficacy of all these biological control agents for the effective management of most of the diseases. And the Royal Commission on Agriculture says that, however, efficient organization which built up for demonstration and propaganda, unless that organization is based on the solid foundation provided by research, it will be merely as house built on land. This is a famous statement given by the Linktigo. With this, I will complete the importance of Trichoderma, a millennium fungus biological agent, by giving acknowledgements to entire the team of Jagirdar for all the PG students and UHS campus Bengaluru for giving this opportunity to give the invited talk. And on behalf of Professor Shamraj Jagirdar, I have done justice to, I think I have given justice to his talk. And thank you very much for one and all.
Thank you, Dr. Gedavare. Very much nice presentation. Audience, any questions, please?
Thank you, sir.
1 or 2 questions, clarifications? Okay. If there are no questions, May I request our anybody else is left, madam? Miss Divya Mishra.
Divya
available?
Mishra.
Divya Mishra, sir,
available?
she's not there. Divya Mishra?
Divya Mishra, please.
No, sir,
if
she's
you are
not
there.
responding. And in the participant list also, she's not there, sir.
Okay.
Sir,
Okay.
excuse me, sir. So I had one question.
Yeah, please go ahead.
Sir, I wanted to ask that in case of soil-borne pathogens like Rhizoctonia and Sclerotium, the main inoculum is the Sclerotia, that is hard structures produced by the pathogen. Can any biocontrol agent degrade these Sclerotia?
So I have shown you. Yes, sir, I'm on line.
Yeah.
Can you hear me, madam?
Yeah, please go ahead. Please go ahead. Yes. Please answer.
Yeah. Just like any hard structures like Sclerotium are very well managed by this trichoderma. Not only trichoderma as a soil inhabitant, but as an endophyte can be used to manage these soil-borne diseases. And there is a successful evidence to showcase that I have shown you one of the slide of potato tuber rot, which is caused by Sclerotium again, wherein it was successfully managed with the help of this Trichoderma harjanum and Trichoderma viridae.
Okay, thank you, sir. Thank you so much.
Any other question?
I have a question. This is Maruti Gowda from the UK. The question is, quite often a concoction of biological control agents and biofertilizer agents are recommended. So because trichoderma actually affects other fungi, is it actually also going to affect?
I think we lost the connection.
Hello, sir.
Thank you, Agdevre.
Dr. Maruti, sir.
Okay, sir.
Okay. Now I request our co-chairman, Dr. Narendra Pa, to give you his remarks.
Thank you, sir. Sir, in total, actually, Maruti Gowda has one question. So, Dr. Maruti, could you please tell us?
Sir, yes, thank you for the opportunity again. I was just wondering, because trichoderma is an antifungal biopesticide, and but it's often recommended with other biofertilizers like nitrogen-fixing bacteria or P-solubilizers and potassium mobilizers and all that. So when they are recommended together, does it also actually affect other bacteria and fungi? What is Dr. Egde's views on that?
See, sir, for any application, such kind of combined applications, before it goes to the field, you will see its compatibility under laboratory conditions. Under laboratory conditions or under the cohort culture studies. So wherever we have used this trichoderma along with the azospirillum or some other organisms, there was no any side effects or there was no any harmful effects. In turn, both together have given more plant growth promotion activities and suppressing the disease.
Okay. So it selectively affects only the pathogens. Is that the answer?
And found compatible with some of the biofertilizers.
Okay. That's good. Thank you. That clarifies my question.
Thank you. Thank you, Agdevre. Thank you. Narendra Pa, please.
Yeah, thank you, sir. Sir, in total, in fact, I'm so much thrilled after a long time. I could hear so many talks, 5 invited talks, and also 5 oral presentations along with the keynote addresses from the 5 people. So I'm very much thrilled. And as everything is there, there are some questions related to alternatives to something like pesticide, carbofuran, maybe other things also. And as Dr. Maruti Gowda and others also put it, consortia, we have started using many of them, and compatibility issues and also the mechanisms and others are being questioned, are being asked. And along with that, Dr. Rao has said mainly about the scale-up registration and the social issues that are related to that. And so there are many questions and also there are some solutions, as Dr. Gurudat Egde has put it, where take the case of trichoderma. If you go with one of the by agents, certainly the work or the reviews, it says that it is a positive thing. And also the stability and other things are the issues in case of maybe the RNA and other cases, but for that they have solutions. And finally, I feel that one the biopesticides maybe the holistic approach, as Dr. Rao put it, commercialization and also in registration and the success story, and do the delivery system, good delivery system, and also work on the quality issues, definitely will pay a way in making up some of these small scale industries, so that we are in the era of like startup programs, and also making the empowering the students as job seekers or and job providers otherwise. And in that way, I feel that one, like taking the example of like talcum powder based trichoderma, or vermicompost based mass multiplied trichoderma, and other these things which creagen and others also they are coming up with. So, and finally, the students are mainly into that. Finally, the endophytes and the products, secondary metabolites, or their characterization and other issues. Certainly, it is good for something that is the innovative research they are doing. But for application things, again, we depend on the old trichoderma, pseudomonas, bacillus, and the other issues. Certainly, we look forward to some of these applications and also the commercialization, popularization, along with the registration and other things, sir. Thank you. Thank you for giving this opportunity.
Thank you.
Yes.
Thank you,
Thank
Dr.
you so much.
Friends, to conclude, I thank all the speakers. 11 speakers were there. I thank all of them, and give some of my views on whatever has been done and my experience as a professor of plant pathology and as the head of the department at UAS Bangalore. What I observed is most of the work is by postgraduate students, and either it is confined to laboratory or greenhouse or microplots. And there are very little work has been done under extensive field trials, and this is required now. Whatever little work the people have contributed, that should be taken forward to field trials and ground truthing, upscaling. All these things has to be done. Secondly, there is no continuity. Once a PG student, MSc students, or PhD student takes up a project, he gets the degree and get out, and continuation is not there. This is another observation, and we should develop awareness among not only farmers but also under scientists, departmental workers, and policymakers. Unless we give awareness for these people, there is no future for bioagents. Please mute others. Please mute. Please mute.
Dr. Palmer.
Krishnappa sir, can you please unmute yourself? Yes, sir. You can proceed.
Either one organism or one crop or one location is the only order of the day, but that is not going to help anything. It's a piecemeal work. So it should be holistic work and continuity, and adoption and upscale should be there. Farmers' awareness should be there, adoption should be there, upscale is there. Anyway, to conclude, it's a very challenging area. It's not an easy job. There are several factors are involved in adoption of bioagents under field conditions. So I request the youngsters to take it forward, and let us see that something will come out. Thank you very much for the organizers, speakers, for your cooperation, patience, and support. Thank you. Over to the organizers, please.
Thank you, sir.
Thank you,
sir.
Dr. Dewappa.
Sir,
Thank you.
I thank the chairman, co-chairman, and the rapporteurs for successfully completing this session. I also thank all the keynote speakers, invited speakers, and the oral presenters for having accepted our invitation and in our internationally conference. Not last but least, I thank all the participants for attending and cooperating to the fullest extent. Thank you, sir. Thank you so much, sir.
Thank you, sir.
In continuation of the previous session, Dr. Selvarajan, sir.
Yeah, I am here.
So we have speakers. Over to you, sir, for continuation of the session. omics and informatics in plant pathosystems, sir. Thank you.
Thank you. Thank you. I have already asked Ashok Kulkarni to share his presentation, and he would be presenting on genomic selections model for blister bar disease tolerance in Carolina natural population. Please, Dr. Ashok Kulkarni.
Yes, sir.
Yeah.
Just three quick checks, sir. My slide is visible, sir?
Very much visible. You can go ahead.
Okay, sir. Okay, just I will take laser. Yeah, my slide is visible. I am audible, and then my pointer is also seen, sir. Okay. Very good afternoon, sir. First of all, I would like to thank all the organizers for giving me this great opportunity. In this presentation, I would like to share my experience on genomic selection model for blister disease tolerance in Casuarina, coming from natural population. This is a collaborative work which was carried out along with COH Bangalore for screening disease tolerant genotypes. I would like to thank Dr. Anjane Reddy and Dr. Devappa for their kind cooperation for conducting this study. Then we all are aware that perennial crop breeding is a big task actually because of the long breeding cycle and then late trait expression and all. So and there is a huge time gap between investment on breeding to deployment of genetically improved material is there. That is a big task actually. And my question here is, can we develop innovative genomic tools to predict a better genotype at the early stage in Casuarina so that breeding cost, breeding cycle, everything can be reduced actually. And these are my contact details actually. In case if you cannot ask or if I cannot convey the necessary message, you can contact me on mail as well as through. I'm always available through WhatsApp and Telegram. This is the outline of my presentation. First few slides, I will talk about distribution of Casuarina, genetic diversity, and then breeding objectives. Next couple of slides, I will be talking on the building genomic selection model for blister disease, and then more importantly, the application of genomic selection and how exactly we can use this in field condition. I will discuss about that. Coming to the distribution of Casuarina, it is almost grown in Casuarina equisetifolia is grown in almost 160 countries, and this was introduced to India in 19th century. In India, almost 8 lakh hectares is being cultivated. Yes, ITC is interested as we are involved in the paper business, so it is one of the raw material for our paper industry. And the Casuarina has a multiple use, right from pulp to scaffolding. Actually, the biggest advantage of Casuarina is it fixes atmospheric nitrogen, and thereby it improves the soil health. So coming to the breeding objectives, we have two important breeding objectives. One is to develop a drought tolerant with high wood yield and pulp yield, which is the most desired trait for our pulp industry. The second most objective is to give the resistant varieties of Casuarina to the farming community. And in this, we have took marker-based approach, wherein we have developed a genomic selection model. And with this, we can predict the phenotype at the early stage itself, and so that we can assure farming community about the better planting material. And the blister bark disease, which is the causal organism, is the Trichosporium vesiculosum. This particular pathogen attacks stem portion of the tree, and this is a typical symptoms wherein bark is pushed outwards due to the excess production of conidia. And we have experienced around 10 to 15% yield loss, and mortality rate is as high as 90%. And some other factors like irrigation, soil pH, and the soil types also play a major role in the disease expression. So the basic requirement of any breeding program is to have a diverse material. When we looked at the existing germplasm, what we had in India was very narrow in genetic diversity. So then we decided to introduce a couple of families of Cunninghamiana and Juveniana from its center of origin, and these were evaluated for blister bark disease. And as you can see here, there is a huge increase in genetic diversity of the germplasm pool after introduction. And this is now I'll briefly explain about the genomic selection model and how this can be used for selecting the better genotype at the early stage. This basically involves phenotyping of disease tolerance was done along with the COH Bangalore, and then we also genotyped with the help of SNP markers, and then later using a statistical model, statistical measures, the prediction model was developed and validated in subsets of training population. So the genomic selection model accuracy depends on the population structure, linkage disequilibrium, and then the trait heritability, and also the effective population size. These are some of the couple of genomic selection publications already published. This basically gives us, confidence on utilization of genomic selection for various traits. And now I will explain how the GS model was developed for Blister disease. We carried out genotyping using diversity array technology, and we observed around 58,461 SNP. After applying a stringent filters, then we got around 32,000 significant SNP markers, and then using the GLA model, we associated around 6,872 markers with the trait of interest. In this case, it is Blister disease. Then we also carried out genome synteny analysis, which also showed that the 84% of the CJ matched on Juguniana markers matched on to the already published Casuarina equisetifolia genome, which has given us the confidence that our genotyping has covered the entire genome. So SNP markers are, this also gives us the confidence that the SNP markers have distributed across the genome. Then coming to the second step of genomic selection model is the accurate phenotyping. As I mentioned earlier, this was done at COH Bangalore campus, and we selected around 62 families of Juveniana, and these were artificially induced with Trichosporium vesiculosum, and then necessary conditions were given for disease expression. And after 6 months, disease severity scale was classified as per the table, and then whatever the phenotypic data was generated through this study was associated with the genotype, which I will explain in my upcoming slides. And the phenotyping study showed that our 40% of the germplasm is tolerant to Blister disease, and almost 26% of the germplasm is highly susceptible. And we also observed that the tolerant clones are showing a high phenol expression. So that is because of the expression of secondary metabolites due to the pathogen infestation. And then we also saw, we did some pathway analysis, and we also realized that these markers are basically categorized into these gene groups. Then this is about the prediction model for Blister disease tolerance. For Blister disease trait, as I mentioned earlier, we observed around 6,872 SNP markers, and out of 32,000 significant SNPs. We used the whatever the phenotypic data which was generated along with COH Bangalore to develop the prediction model. And these are the families which are showing high breeding value, basically GBV. So which we can conclude that these are tolerant for Blister disease. And this was further validated in the validation population, wherein we found around the prediction accuracy around 0.76. This gives an assurance that the whatever we have developed the Blister disease tolerant model, so that can be used for predicting the better genotypes at the early stage. And then further, the model was further validated in second generation material, which was derived from the clonal seed orchid. So seedlings were genotyped at the early stage, and then the breeding values were estimated. Parallelly, these seedlings were planted in various locations for observing the disease expression. So using the developed model, we observed that 13 families are tolerant to Blister-Buck disease, and the predicted data was compared with the field data, and we observed that 80 to 90% of the the similarity with the predicted values. This again gives us the confidence on genomic selection for early selection of superior genotype with desired set. And I will conclude my talk saying that we have developed first prediction model in Casino for Blister-Buck disease with accuracy of 0.76. Yes, ultimately through genomic selection model we can achieve a genetic gain, which is now possible through these new tools which are developed in genomics. And this is our team, and of course this has scientists from different domains apart from silviculture, and this is our lab at Bangalore. It's a state-of-art lab, and we have got all the basic facilities for conducting the genomic studies. So thank you and for your patience, and I'm open for any kind of suggestions and feedback. Thank you very much, sir.
Yeah, now the presentation is open for discussion. Any questions, please? Any questions? Okay, it's a really wonderful work using dot sequencing technology for genotyping and phenotyping and associating with this, and it's really a very wonderful technology, and which we have applied for this Casverina, which is used in the paper industry. It's very good work, Dr. Kulkarni, and I congratulate you and appreciate you for this a wonderful presentation. And next, we will move down to the next speaker, Mr. Amoga Varsha. Mr. Amoga Varsha, are you there? I was keep on calling, but he was not there. I was chatting and inviting him. Oh, you are there?
Not there.
Not there. No, yeah, I was keep on asking everyone. Anyhow, somehow we missed it because he would have thought that we will not give him chance probably. Anyhow, we have to adjust the time, and now I request our co-chairman to give his comments, Dr. Dhananjaya. Dr. Dhananjaya. Dr. Dhananjaya, you are a co-chairman of this.
Sir, Dhananjay, sir, can you please unmute yourself?
Sir, I am audible? Yeah, you are very much audible. Yeah.
Okay, because there is some problem here. I am not able to hear properly. There is audio issues there, and even videos also continuity is not there.
Oh, okay.
Lot of disruptions are there.
Okay.
Anyhow, I thank the organizers for providing me an opportunity as co-chairman of the session, and really the keynote addresses of this session gave a lot of new information to our student fraternity and academicians, and also scientific this thing of our country also in specific and in general to all those who are involved in the latest research in plant pathology using omics and other informatic bioinformatic fields. But the thing is, considering the infrastructure at some important laboratories of our country, still there is scope that we have to ramp up our infrastructure to match the research work being done at other like developed countries' laboratories. Otherwise, we will be lagging behind in really coming out with at par research output that other laboratories are doing. Though our manpower, there is no lack of this thing, expertise and capabilities. Only thing is because of maybe the inadequate infrastructure. With the frontier area of research, there is still scope for like improving our capabilities in the infrastructure, so that the outcome of these all omics and informatic research projects can be put into practical use, because we know that in the case of rice bacterial blight disease and also the rice blast disease, they are controlled by many genes actually. There are many races prevailing, many pathogens are prevailing, and even identifying one or two prominent QTLs for managing these diseases. We took a lot of time to come out with varieties and like genotypes for commercial cultivation. So the information on this, whatever the latest frontier field of science, that omics and information technology information, to put it into practical use, there's still like we have to go a long way so that we can hasten the process of crop improvement and also come out with resistant cultivars for immediate commercial utility. So with these few words, I thank the organizers, and especially our chairman really managed the whole affairs in a very efficient and effective way. And he was very critical also in between the presentations. I thank the organizers for providing me an opportunity to share my few words about the conference in general, particularly about the session in which I got an opportunity to co-chair along with Dr. R. Selvirajan. Thank you, organizers, and also thank you, chairman, rapporteurs. Thank you, one and all.
Thank you. Thank you, Dr. Dhananjaya. Thank you, Dr. Dhananjaya. Now, I would just brief my comments on this omics and bioinformatics in plant pathology session. And yes, our co-chairman said it was a very wonderful talks from the international scientists from USA and also from NCL, our India. Dr. Murali Raghavendra Rao, an alumni of GKVK, gave a wonderful talk on bioinformatics, and he was telling how to use this cloud and other like microbiome and other way of how to use the big data analysis and how to use the pipeline based analysis, and how to he also ensured that safetiness of the data which we share in the cloud. And now for in IoT based managements, we need to handle very big data, and those data without cloud support it's impossible to handle. So he gave a very strong support and confidence to make our scientists to use effectively the cloud. That was very interesting talk by Dr. Murali Raghavendra Rao. The next, Hanu Pappu, he gave a wonderful talk on the tomato spotted wilt virus, and tomato spotted wilt virus being a new to India. That is, though we have got a groundnut bud necrosis virus in general, but recently the TSW has been detected using even nanopore sequencer in our laboratory. Also, we have done with the Dr. Krishna Reddy's collaboration and other thing. So what I mean to say is TSWV is going to be a very big problem in India also, and he gave such a wonderful outline of the work which what he was doing for the over one and a half decades on TSWV in Pullman, Washington State University. So it was very thought-provoking talk by Dr. Pappu, and I hope all our students and staffs and faculties have enjoyed his talk, and the way he took from starting from inoculation to the omics approach application and differentiating the resistant and susceptible, and the pathogen point of view and the host point of view, and how CRISPR can be applied to make the susceptible, high yielding susceptible variety to make the resistant variety by handling the susceptible gene. Means editing the susceptible gene and all he was killing. It's really thought-provoking. And coming to the Narendra Kado, it was a very, very wonderful talk by on chickpea fusarium interactions, and starting from he took transcriptomics, proteomics, and metabolomics, all three put together, and he brought out such a wonderful outcome. And he related really all the approaches of omics, and that was really a good presentation for all our students who could understand that the interaction has to be studied not only at transcriptomic level but also metabolomics level that determines the disease defense. And coming to there were 5 oral presentations, but there were 4 presented. One by Dr. Raghavendra Gunnaya, that is on Xanthomonas saxonopodis punicae. That's a very, very serious disease, especially for Karnataka. And he told that bacteriophages, which is plenty, which can be utilized as a biopesticide. That's a very good one, and a further study in that would lead into a eco-friendly technology in future. So that was a wonderful talk by Dr. Raghavendra. And coming to the Dr. Pramesh, he gave again a very good talk on molecular characterization of comparative genomics and mating type locus analysis of Aspergillosis variants from the rice infecting. So that was really wonderful talk because he started from the culture morphology. Then again, he came to the molecular analysis of those cultures and the plates, what he has shown in the presentation. It was so wonderful. And such are the studies are very much needed to have a very deep understanding about a pathogen. So really, it was a wonderful talk, and 10 minutes was not sufficient for these people, but it was very, very thought-provoking oral presentations. And coming to the Pernospora plant genus draft genome sequence from that is medicinal and aromatic Directorate of Medicinal and Aromatic Plants. That was also very good by Nagaraj Reddy. And I think the draft genome is yet to be submitted, but it was a wonderful piece of work on this particular crop he has done on Pernospora. That's a very good work what he presented. And finally, Kulkarni, he gave a very nice talk on Casuarina, and he was telling about 8 lakh hectares in India it is being grown. And banana also 8 lakh hectares only we grow, and we are number 1 fruit crop in India. And Casuarina also very important. It's equally grown in 8 lakh hectares of area. And a deep research in this blister bar disease and a selection model based on the dart seq technology was very good and really wonderful. These things have to be applied in other crops. This type of dot technology has to be applied even with pomegranate, bacterial blight, and other diseases. The important diseases which really daunting our Indian agriculture, like sugarcane, red rot, and there are many diseases like Fusarium, tropical race for Fusarium, banana. There are many important diseases for which we can apply this these dot-based technologies to go for a genome-based selection of the germplasms and other things. And with this, I would say that this omic approach and this bioinformatics and informatics in plant pathology theme had a very wonderful presentations, and it's all thought-provoking, and I hope that it was useful to each and every participants. And I thank the organizers. Just with 100 rupees, we could able to listen to the international speakers. 100 rupees registration, and all the students get a opportunity to attend this section. Otherwise we have to pay 10,000 rupees plus we have to go to and fro, stay, everything 25,000. Every person has to spend money to attend such a international conference. But now we could have a wonderful conference here, and I congratulate Dr. Devappa, Sankarappa, and all the team, the entire team conducting this e-conference. And in fact, I also attended some of the most of the presentations, mainly from abroad and all. From Australia, what she presented biopesticide, Dr. Nina Mittal, and it was very interesting and all. So I attended all those sessions, and I am very happy, and I can once again congratulate. And I hope that our rapporteurs would have taken the recommendations from these points what we presented. So with this, I conclude. I thank once again all the organizers for giving me the opportunity to chat. Thank you very much. Thank you very much. Thank you, Dr. Dhananjaya. Thank you.
Thank you, sir.
So over to organizers.
So I thank the chairman, co-chairman, and the rapporteurs for having given their valuable comments. compliments and suggestions to all the speakers and for the successful completion of the session. Further, I thank all the keynote speakers, oral and the oral speakers for their wonderful presentation. I thank profusely all the participants for their patient hearing. We have come to the end of the third day of our conference. We shall be starting our conference tomorrow at 9:30 a.m. I request all the participants to follow the link which will be provided tonight and make our international e-conference a grand success. Thank you. Thank you one and all.
ಇವಾಗ ಬನ್ನಿ ಕ್ಲೋಸ್ ಮಾಡ್ತೀನಲ್ಲ. ಹಾ
ಅಂದ್ರೆ ನಾನು ನೋಡಕ್ಕಾಗಲ್ಲ ನನಗೆ ಈಗ ನೋವಾಗಲ್ಲ ಈಗ ನಮ್ಗೆ ಕೆಲಸ
ಇನ್ನೊಮ್ಮೆ ಸಲ್ಲಿಸ್ತೀರಾ? ಇನ್ನೊಮ್ಮೆ ಸಲ್ಲಿಸ್ತೀರಾ? ಓಕೆ ಅದು ಮಾಡಿದ್ರಾ? ಲೈವ್ ಸ್ಟ್ರೀಮಿಂಗ್ ಅದು ಇದು ಸೆಕೆಂಡ್ಸ್ ಲೈವ್ ಸರ್ ಇಲ್ಲ ಐತ್ರಿ ಕೆಂಪ್ರಿ ಎಲ್ರಿ ಸರಿ ಲಾಸ್ಟ್ ಸ್ಕ್ರೀನ್ ಲಾಸ್ಟ್ ಕ್ರೀ ಚಲೋ ಐತ್ರಿ ಕೆಂಪು ಮಂಡಲ ಯಾವ್ದು ಲೈವ್ ತೋರ್ಸಾಕತ್ರಿ ಇದಲ್ರಿ ಸರ್ ಇದ್ರೋಗ ಐತ್ರಿ ಲಾಸ್ಟ್ ಲೈವ್ ಸ್ಟ್ರೀಮಿಂಗ್ ಐತಲ್ರಿ
ಈಗ ತಡವಾಗಿ 5 ತಿಂಗಳು
ಐತ್ರಿ ಸರ್ ಇಲ್ರಿ ಸರ್ ಇಲ್ಲ ಐತ್ರಿ ಹಾ ಎಂಡ್ ಆಫ್ ಲೀವ್ ಮೀಟಿಂಗ್ ಅಂತ ಐತ್ರಿ ಎಂಡ್ ಮೀಟಿಂಗ್ ಏನ್ರಿ